Inside every cell, there are enzymes performing a trick that classical physics insists should be impossible, moving particles through barriers rather than over them, as though the rules that govern ordinary matter do not quite apply when life is doing its quiet, necessary work. This is the strange territory where biology and quantum mechanics meet, a place most of us never think to look, and yet it hums along constantly within us, beneath awareness, beneath breath. If these kinds of slow wanderings through science bring a certain stillness, subscribing might be a gentle way to find more of them. Tonight the subject is catalysis at the quantum scale, the way enzymes seem to cheat time and energy, the tunneling of protons through impossible walls, and how, in understanding this, we glimpse something profound about what it means for matter to be alive. The story begins here, in the space between chemistry and mystery. And so we come now to a rather quiet corner of enzymology, where something quite extraordinary happens, though it happens so gently and so persistently that you might almost miss it if you were not paying careful attention. This is the story of how hydrogen atoms, those smallest and lightest of all atomic participants in biological chemistry, manage to do something that, according to the classical rules of chemistry, they really should not be able to do at all. They pass through barriers rather than over them, like a gentle breeze moving through a hedge rather than a bird flying above it, and in doing so, they allow the chemistry of life to proceed at rates that would otherwise be quite impossible at the modest temperatures where living things conduct their business. To understand why this matters, and why it took so many careful years of experimental work to demonstrate that it was happening, we need to first consider what chemists call the Arrhenius equation, which is a rather beautiful and simple relationship that describes how quickly chemical reactions proceed at different temperatures. The equation tells us that the rate of a reaction depends on the temperature, certainly, but it depends even more critically on something called the activation energy, which is the height of the energy barrier that the reacting molecules must climb over in order to transform into products. You can picture this barrier as a gentle hill between two valleys, where the reactants sit in one valley and the products rest in the other, and the only way to get from one valley to the other, according to classical chemistry, is to climb up and over the hill that separates them. The Arrhenius equation tells us that the higher this hill, the slower the reaction will be, because fewer molecules at any given moment will have enough energy to make it over the top. Now, here is where enzymes present us with something of a puzzle that chemists and biochemists had noticed for quite some time but had not fully reckoned with. Enzymes accelerate chemical reactions by factors that can reach into the millions, sometimes the billions, and they do so at body temperature, which is to say at around 37 degrees Celsius for humans and similar temperatures for other warm-blooded creatures. The classical explanation for how enzymes achieve these remarkable accelerations is that they lower the activation energy, that they somehow smooth down the hill between reactants and products so that it becomes easier to climb over. And this is true, as far as it goes, but when researchers began to look very carefully at certain enzyme-catalyzed reactions, they found that the rates were faster than even this lowered barrier could account for. Something else was happening, something that the Arrhenius equation, in its classical form, could not quite explain. The person who pursued this puzzle most persistently, and who eventually demonstrated its solution with beautiful experimental clarity, was Judith Klinman, a biochemist at the University of California, Berkeley, whose career has been devoted to understanding the intimate details of how enzymes work at the atomic level. Klinman began her investigations into this question in the 1970s, when quantum mechanical effects in enzyme catalysis were considered by most biochemists to be either negligible or non-existent. The prevailing view was that while quantum mechanics certainly governed the behavior of electrons, which are very light particles indeed, the heavier nuclei of atoms like hydrogen, carbon, and oxygen could be safely treated as classical objects that obeyed the familiar laws of Newtonian mechanics. Klinman suspected that this view might be incomplete, particularly for hydrogen, which is after all the lightest of all atoms and might therefore retain some of its quantum character even when bonded within a large biological molecule. The phenomenon that Klinman was investigating is called quantum tunneling, and it is one of the stranger predictions of quantum mechanics, though it has been thoroughly verified in countless physical systems. Tunneling occurs when a particle passes through an energy barrier rather than over it, appearing on the other side without ever having had enough energy to climb to the top. This is possible because quantum particles do not have precise positions the way classical objects do. Instead, they are described by wave functions that spread out in space, and if the barrier is thin enough, and the particle is light enough, then the wave function can extend through the barrier and give the particle a small but real probability of being found on the other side. For heavy particles, this probability is vanishingly small, but for light particles like electrons, it can be quite significant. The question was whether hydrogen, which is heavier than an electron but still very light by atomic standards, could tunnel through the barriers encountered in enzyme-catalyzed reactions. The experimental approach that Klinman developed to test this hypothesis was based on something called the kinetic isotope effect, which is a method of using the different masses of isotopes to probe the mechanism of a chemical reaction. Hydrogen has two heavier isotopes, deuterium and tritium, which have 1 and 2 extra neutrons respectively in their nuclei. Because these isotopes have different masses, they behave differently in chemical reactions, moving more slowly and tunneling less efficiently than ordinary hydrogen. By comparing the rates of a reaction when hydrogen is replaced by deuterium or tritium, researchers can learn a great deal about whether and how hydrogen transfer is involved in the rate-limiting step of the reaction. In classical chemistry, the kinetic isotope effect has well-defined limits. When a hydrogen atom is transferred in the rate-limiting step of a reaction, replacing it with deuterium typically slows the reaction by a factor of about 2 to 7, depending on the details of the mechanism. This is because deuterium, being heavier, has a lower vibrational frequency in its bonds, which affects how easily it can reach the transition state at the top of the energy barrier. A ratio of 7, comparing hydrogen to deuterium, is about the maximum that classical theory predicts for a simple hydrogen transfer reaction. If tunneling is involved, however, the isotope effect can be much larger, because the heavier isotopes tunnel much less efficiently than hydrogen, and the rate difference between them becomes dramatically amplified. What Klinman and her colleagues found, over many years of careful experiments, was that certain enzymes showed kinetic isotope effects that were far larger than the classical maximum. In her work on the enzyme alcohol dehydrogenase, which catalyzes the removal of hydrogen from alcohol molecules. She observed isotope effects with ratios that reached 50, 60, even 80 when comparing hydrogen to tritium. These values were simply impossible to explain without invoking quantum tunneling. The experiments published in 1989 represented a breakthrough moment, providing the clearest evidence yet that tunneling was not just a theoretical curiosity but an actual mechanism by which enzymes achieved their remarkable catalytic speeds. But this was not the end of the story, not by any measure, because understanding that tunneling occurred was only the first step toward understanding how it occurred, and how enzymes might have evolved to exploit this quantum mechanical phenomenon. Klinman continued her investigations for decades, refining her experimental techniques and developing theoretical frameworks to explain her observations. One of the key pieces of evidence that helped establish the quantum nature of hydrogen transfer was the discovery of temperature-independent kinetic isotope effects in certain enzymes. In classical chemistry, the isotope effect depends on temperature, because temperature affects how molecules distribute themselves among different energy states. But when tunneling dominates the reaction mechanism, the isotope effect can become independent of temperature, or nearly so, because the particles are passing through the barrier rather than over it, and the barrier height becomes less relevant. A particularly striking example of this came in 2006, when a paper published in Nature examined the enzyme soybean lipoxygenase, which catalyzes the addition of oxygen to fatty acid molecules. The researchers, building on Klinman's work and that of others, showed that in this enzyme, quantum tunneling accounted for an extraordinary 99% of the hydrogen transfer reaction. This was not a minor correction to classical behavior but an almost complete replacement of it. The enzyme had apparently evolved to exploit tunneling so effectively that the classical pathway, the one that goes up and over the energy barrier, had become almost irrelevant. To explain these findings, theorists developed what are sometimes called Marcus-like models, building on the theoretical framework that Rudolf Marcus had created to describe electron transfer reactions. These models incorporated quantum tunneling into a broader picture of enzyme catalysis, describing how the enzyme creates a pre-organized environment that brings the donor and acceptor atoms into close proximity and optimal alignment, reducing the width of the barrier through which the hydrogen must tunnel. The models also described how protein motions, the subtle vibrations and fluctuations of the enzyme structure, might modulate the tunneling probability by transiently compressing the barrier and increasing the tunneling rate. It would be a gentle understatement to say that these ideas were not immediately accepted by everyone in the biochemistry community. Many researchers were skeptical that quantum effects could play such a significant role in the warm, wet, crowded environment of a biological cell, where thermal fluctuations and molecular collisions might be expected to destroy the delicate quantum coherences that tunneling requires. There were long debates at scientific meetings, papers published arguing for and against the importance of tunneling, and a gradual accumulation of evidence that eventually shifted the consensus. The controversy was never acrimonious, merely persistent, as is often the case when a new idea challenges established ways of thinking about a familiar subject. What emerged from these decades of research was a picture of enzyme catalysis that is richer and more nuanced than the classical view had suggested. Enzymes do indeed lower the activation energy for the reactions they catalyze, bringing reactants together in precisely oriented configurations and providing chemical groups that stabilize transition states. But some enzymes also exploit quantum tunneling, creating conditions where hydrogen atoms can pass through barriers rather than over them, achieving reaction rates that would be impossible if chemistry obeyed only classical rules. The fact that biology has discovered and optimized these quantum mechanical tricks through the slow patient process of evolution is really quite remarkable, though it happens every moment in every cell of your body without any fuss or fanfare. Judith Klinman's quest, which began in the 1970s and continued for more than 4 decades, exemplifies the kind of patient, persistent inquiry that characterizes the best scientific work. She asked a question that many thought was already answered, or not worth asking, and she pursued it with careful experiments and rigorous analysis until the answer became clear. The enzymes that catalyze hydrogen transfer reactions in your body, right now, as you listen to these words, are using quantum tunneling to do their work, allowing hydrogen atoms to take impossible jumps through energy barriers, keeping the chemistry of life running smoothly at temperatures far lower than those required for the same reactions to proceed in a test tube without enzymatic assistance. And so we find that the boundary between quantum mechanics and biology, which might once have seemed quite sharp and well defined, is actually rather soft and permeable, with quantum effects reaching up into the warm realm of living chemistry and making themselves essential to processes that had seemed purely classical. The hydrogen atoms that tunnel through enzyme barriers are very small, and the barriers are very thin, and the whole process happens in times measured in femtoseconds, which are millionths of a billionth of a second. But these tiny events, happening in countless enzymes in countless cells, add up to the smooth continuous chemistry of life, the gentle transformations of molecules that keep us breathing and thinking and dreaming. It is a reminder, perhaps, that the universe operates according to its own rules, which do not always match our intuitions, and that biology has had a very long time, measured in billions of years, to discover and exploit those rules in ways that we are only now beginning to understand. And now we turn our attention to one of the most fundamental processes on Earth, the quiet and unceasing work of photosynthesis, which has been converting sunlight into usable energy for billions of years, long before there were eyes to witness the green of leaves or the shimmer of algae across still water. This is a process so familiar that we might take it for granted, yet within its molecular machinery there appears to be something quite unexpected, something that connects the warm, wet world of biology to the strange and delicate realm of quantum mechanics, and understanding this connection requires us to look very closely at what happens in the first few moments after light strikes a photosynthetic organism, when energy must travel from where it is captured to where it can be used. The story we are about to explore begins with a particular protein complex that has become famous in the world of quantum biology, known as the Fenner-Matthews-Olsen complex, or simply FMO, which was first characterized in the early 1970s by Roger Fenner and Brian Matthews working with the crystallographer John Olsen, and which has since become one of the most studied light harvesting systems in all of biochemistry. This complex is found in green sulfur bacteria, organisms that live in places with very little light, such as the deep layers of stratified lakes or the sediments of hot springs, and which have evolved to capture and use almost every photon that reaches them, making them remarkably efficient at the business of photosynthesis. The particular species most often studied is called Chlorobaculum tepidum, a warm-loving bacterium that was originally isolated from hot spring microbial mats in New Zealand, and which grows best at temperatures around 48 degrees Celsius, making it quite comfortable in conditions that would be rather unpleasant for most other forms of life. The FMO complex serves as a kind of wire or conduit, receiving energy from the large light-harvesting antenna structures of the bacterium and transferring that energy to the reaction center where the actual chemistry of photosynthesis takes place. It is a relatively small protein, containing 7 or 8 bacteriochlorophyll molecules held in a precise arrangement by the surrounding protein scaffold, and the question that interested researchers was how this energy transfer could be so extraordinarily efficient, approaching 99% in some measurements, when the physics of random energy transfer would seem to predict much greater losses. If energy were simply hopping from one chlorophyll molecule to the next in a random walk, bouncing this way and that until it happened to find its way to the reaction center, one would expect significant energy to be lost along the way, dissipated as heat or re-emitted as fluorescence, yet the bacteria managed to guide almost all of the captured energy to precisely where it needs to go. In 2007, a team of researchers led by Graham Fleming at the University of California, Berkeley, published a paper in the journal Nature that would spark a new field of investigation and generate considerable excitement and controversy in equal measure. The lead author of this paper was Gregory Engel, then a postdoctoral researcher in Fleming's laboratory, and the technique they employed was something called two-dimensional electronic spectroscopy, which allows scientists to observe the movement of energy through molecular systems with extraordinary precision. This method uses sequences of ultra-short laser pulses, each lasting only a few tens of femtoseconds, to excite the sample and then record how the excitation evolves over time. And by analyzing the resulting signals, one can build up a detailed picture of the energy transfer pathways within the system. What Engel and his colleagues observed was something that no one had quite expected. When they looked at the signals from the FMO complex, they saw a pattern of oscillations, a kind of beating that persisted far longer than anyone had anticipated. These oscillations, which came to be called quantum beating signals, are a signature of quantum coherence, meaning that the energy excitation was not behaving like a classical particle hopping randomly from site to site, but was instead existing in a superposition of states, spread across multiple chlorophyll molecules simultaneously. The remarkable finding was that these coherent oscillations lasted for approximately 660 femtoseconds at physiological temperatures, which is to say at temperatures close to those experienced by the living bacteria, around 77 Kelvin in the initial experiments and later confirmed at higher temperatures closer to ambient conditions. To appreciate why this finding was so surprising, one must understand that quantum coherence is generally considered to be extremely fragile. In most laboratory settings, achieving and maintaining quantum superpositions requires cooling systems to temperatures near absolute zero, isolating them from any environmental disturbance, and working with carefully controlled conditions that bear little resemblance to the warm, wet, noisy interior of a living cell. The prevailing wisdom had been that biological systems were simply too hot and too busy to sustain quantum effects for any meaningful length of time, that decoherence would destroy any quantum superpositions almost as soon as they formed, and that biology, whatever its other wonders, operated firmly in the classical world. The Berkeley experiment seemed to challenge this assumption, suggesting that somehow the FMO complex was able to protect quantum coherence from the environmental noise that should have destroyed it almost immediately. The theoretical implications of this finding were intriguing. If energy could exist in a coherent superposition across multiple sites, it could effectively explore all possible pathways to the reaction center simultaneously, rather than taking a random walk through the system. This is sometimes described using the metaphor of a quantum search algorithm, where the coherent excitation samples many routes at once and interferes constructively along the most efficient path while interfering destructively along wasteful detours. The result would be energy transfer that is both faster and more efficient than anything achievable through classical means, and this might explain how green sulfur bacteria are able to achieve their remarkable near-perfect efficiency in capturing and using light energy. The protein scaffold surrounding the bacteriochlorophyll molecules appears to play a crucial role in this process, creating what some researchers have described as a kind of protected environment or quantum cradle for the coherent energy transfer. The arrangement of amino acids in the protein creates a specific dielectric environment around each pigment molecule, tuning the energy levels and coupling strengths in ways that may facilitate coherent energy flow. There is also evidence that the protein may provide a degree of rigidity that reduces certain types of thermal fluctuations while still allowing others, and some theoretical work has suggested that this balance between order and disorder might actually enhance rather than destroy quantum coherence, a phenomenon sometimes called environment-assisted quantum transport or, more colloquially, the Goldilocks effect. The publication of the Nature paper generated considerable attention both within and beyond the scientific community, and the idea that photosynthesis might involve quantum mechanics captured the imagination of many who had not previously given much thought to the inner workings of chloroplasts and bacterial energy systems. However, as is proper in science, the initial excitement was followed by careful scrutiny and attempts to replicate and extend the findings, and over the following decade a more nuanced picture began to emerge. In 2018, a series of studies began to question whether the observed coherences were truly quantum mechanical in the sense that had been initially proposed. Research groups including those led by David Jonas at the University of Colorado and Paul Broomer at the University of Toronto pointed out that the beating signals observed in two-dimensional spectroscopy could arise from purely vibrational effects, from the mechanical oscillations of the molecular structure rather than from electronic quantum coherence. The distinction is subtle but important. Quantum coherence between electronic states would allow for the kind of superposition-based exploration of pathways that had been proposed as the explanation for high efficiency, while vibrational coherence, though still a quantum phenomenon in the strict sense, would have different implications for the energy transfer process and might not provide the same kind of enhancement. The debate that followed has been technical and detailed, involving careful analysis of spectral signatures and timing sequences, and it continues in various forms to this day. Some researchers argue that while purely electronic coherence may be shorter-lived than initially thought, there is still evidence for coherent coupling between electronic and vibrational degrees of freedom that may play a functional role. Others maintain that the efficiency of biological light harvesting can be adequately explained by classical or semi-classical models without invoking long-lived quantum coherence. What everyone agrees upon is that the protein environment is exquisitely tuned to facilitate efficient energy transfer, whether that tuning involves protecting quantum coherence, providing directed classical pathways, or some combination of both. It is worth noting that the original Berkeley experiments were conducted at quite low temperatures initially, 77 Kelvin, which is the temperature of liquid nitrogen and far colder than any living bacterium would experience. Later experiments showed coherence persisting at higher temperatures, but the question of whether these effects are present and functional under genuinely physiological conditions remains a subject of investigation. Chlorobaculum tepidum lives at temperatures around 40 to 50 degrees Celsius, and while some coherent signatures have been observed at elevated temperatures, they are generally shorter-lived and less pronounced than those seen in the cryogenic experiments. What we can say with confidence is that the FMO complex and other light harvesting systems are extraordinarily well designed for their purpose, channeling energy with an efficiency that human engineers can only admire and attempt to imitate. Whether this efficiency arises from quantum coherent effects, from carefully structured classical dynamics, or from some interplay between the two, the result is a system that has been refined over billions of years of evolution to extract maximum value from every photon of light that reaches it. The green sulfur bacteria living in the dim waters of stratified lakes have no need to understand the physics of their own existence, yet they have evolved solutions to problems of energy transfer that continue to challenge our understanding and inspire new directions in research. The controversy over quantum effects in photosynthesis has, if nothing else, demonstrated the importance of looking closely at the assumptions we bring to our investigations of nature. For many years, the assumption that biology was too warm and too wet for quantum effects led researchers to ignore possibilities that now seem worthy of careful consideration. Whether or not long-lived quantum coherence turns out to be central to photosynthetic efficiency, the investigation has opened new avenues of inquiry and revealed previously unappreciated subtleties in the interactions between light, matter, and the complex molecular structures that living systems have evolved. As you rest here listening to these words, you might reflect gently on the fact that the oxygen in the air around you, the oxygen your body is quietly using at this very moment, was produced by photosynthetic organisms not so very different from the green sulfur bacteria we have been discussing. The quantum mechanics of energy transfer, whether coherent or classical, underpins the chemistry that sustains almost all life on Earth, and there is something rather peaceful in knowing that such fundamental processes continue without interruption, in oceans and forests and the smallest of ponds, while you drift towards sleep. The 660 femtoseconds of coherence observed in those Berkeley experiments is an unimaginably brief interval by everyday standards, roughly the time it would take light to travel the width of a human hair, yet within that fleeting window the energy of a captured photon is guided with remarkable precision toward the molecular machinery that will put it to use. And whether we understand every detail of how this happens or not, the process itself continues, as it has for billions of years, the green quantum machines of photosynthesis quietly turning starlight into life. Let us begin with a quiet moment in the late 19th century, in a German laboratory where the air smelled of reagents and the light came through tall windows. falling across the workbenches where a chemist named Emil Fischer was thinking about shapes. It was 1894, and Fischer had been studying enzymes, those mysterious substances that seemed to guide chemical reactions in living things with an almost knowing precision. He had noticed something that struck him as quite important, something that would shape how scientists thought about enzymes for the next century and beyond. The enzymes he studied seemed to be particular about their partners, choosing only certain molecules to work with while ignoring others that seemed almost identical. This selectivity reminded him of something mechanical, something familiar, and so he proposed what came to be called the lock and key hypothesis, suggesting that an enzyme and its substrate fit together like a key sliding into a lock, their shapes complementary in a way that allowed them to interact while excluding all other molecules that did not possess the correct geometry. This was a comforting idea, really, the notion that the specificity of enzymes could be explained by something as tangible as shape, as the simple mechanical matching of three-dimensional forms. Fischer's lock and key model gave scientists a way to visualize what was happening at scales too small to see, and it proved remarkably useful for many decades. But as with many ideas that seem complete and satisfying at first, there were aspects of enzyme behavior that the lock and key hypothesis could not quite explain. For if an enzyme and its substrate fit together perfectly, like a key in a well-made lock, then why would the enzyme ever let go of the substrate once the reaction was complete? And more troublingly, how did the enzyme actually make reactions happen faster, rather than simply holding onto molecules without changing them? The lock and key model described recognition beautifully, but it left the question of catalysis itself largely unanswered, like a story that introduces its characters but never quite gets around to telling you what they do. More than 50 years would pass before this puzzle found its most influential answer, and that answer came from a man whose name you may know from other contexts, a chemist of extraordinary range whose work touched nearly every corner of his field. Linus Pauling, working in the 1940s at the California Institute of Technology, proposed something rather counterintuitive about how enzymes work. In 1946, he suggested that the key to enzyme catalysis lay not in how tightly enzymes bind to their substrates in their normal, stable form, but rather in how tightly they bind to something called the transition state. To understand this, we need to think for a moment about what happens during a chemical reaction, and you may find it helpful to imagine a ball rolling over a hill. The substrate, the starting material of a reaction, sits in a valley on one side, and the product, the end result, sits in a valley on the other side, and between them rises a hill that the ball must climb over. The top of this hill represents the transition state, a fleeting, unstable arrangement of atoms that exists for only the briefest of moments as the reaction proceeds. Pauling's insight was that enzymes must have evolved to bind this transition state more tightly than they bind either the substrate or the product. By stabilizing the transition state, by making that unstable arrangement of atoms feel more comfortable and less energetically costly, the enzyme effectively lowers the height of the hill. The ball does not need to climb as high to get over to the other side, and so the reaction proceeds more quickly. This was a profound reconceptualization of enzyme function, shifting the focus from static shape matching to dynamic stabilization of an ephemeral molecular arrangement. The enzyme, in Pauling's view, was not merely a passive host but an active participant, reshaping the energetic landscape of the reaction to make the difficult easy. While Pauling was thinking about transition states, another piece of the puzzle was being assembled in Berlin, where two scientists had worked together more than 3 decades earlier to establish the mathematical foundations of enzyme kinetics. In 1913, Leonor Michaelis and Maud Menthen published a paper that would become one of the most cited in all of biochemistry, describing what is now known as the Michaelis-Menten equation. Menthen was a Canadian physician and biochemist who had traveled to Berlin to work with Michaelis, and together they studied how the rate of an enzyme-catalyzed reaction depends on the concentration of the substrate. Their equation, which relates reaction velocity to substrate concentration through two constants now called kilometer and Vmax, provided scientists with a quantitative framework for describing enzyme behavior. The kilometer, or Michaelis constant, tells you something about how tightly an enzyme binds its substrate, while Vmax describes the maximum rate at which the enzyme can work when it is fully saturated with substrate. This mathematical description allowed researchers to compare enzymes to one another, to understand how changes in conditions affected catalysis, and to begin unraveling the mechanisms by which individual enzymes performed their tasks. It was careful, methodical work, the kind of work that builds foundations even if it does not make headlines, and it remains essential to enzymology to this day. As the decades passed and more was learned about enzyme structure, scientists began to realize that Fisher's lock and key model, while elegant, was perhaps too rigid in its conception. Enzymes were not static objects, it turned out, but flexible molecules that could change their shape in response to binding their substrates. This realization was formalized in 1958 by Daniel Koshland, working at Brookhaven National Laboratory on Long Island, who proposed what he called the induced fit model. Koshland suggested that when a substrate approaches an enzyme, the enzyme does not simply wait passively for the substrate to fit into a preformed pocket. Instead, the enzyme changes its shape, adjusting its conformation to better accommodate the substrate and to position the reactive groups in exactly the right arrangement for catalysis to occur. The lock, in other words, reshapes itself around the key. This model explained observations that the lock and key hypothesis could not, such as why some molecules that seem to have the right shape to fit into an enzyme's active site nonetheless failed to trigger catalysis. The induced fit model suggested that proper binding required not just shape complementarity but the correct induction of conformational changes, a dynamic handshake between enzyme and substrate rather than a static meeting of predetermined forms. With these three conceptual frameworks in place, lock and key specificity, transition state stabilization, and induced fit dynamics, scientists felt they had a reasonably complete picture of how enzymes worked. These ideas were taught in classrooms and written into textbooks, and for many practical purposes they served admirably well. But there remained a quiet problem, a nagging discrepancy that would not quite go away no matter how carefully the theories were applied. This problem came to be known as the rate enhancement problem, and it concerned the sheer magnitude of the accelerations that enzymes achieve. Consider, if you will, an enzyme called arotidine decarboxylase, which participates in the synthesis of the nucleotide building blocks that make up your DNA and RNA. This enzyme catalyzes a reaction that, without the enzyme present, would take an almost unimaginably long time to occur. In the 1990s, a biochemist named Richard Wolfenden, working at the University of North Carolina at Chapel Hill, set about quantifying just how slow this uncatalyzed reaction really was. What he found was rather startling. The reaction that arotidine decarboxylase catalyzes, in the absence of the enzyme, has a half-life of approximately 78 million years. That is to say, if you were to take a collection of arotidine molecules and leave them in water without any enzyme present, it would take 78 million years for half of them to undergo this particular chemical transformation. And yet, in the presence of the enzyme, the reaction proceeds essentially instantaneously on biological timescales. The rate enhancement, when calculated carefully, works out to a factor of approximately 10 to the 17th power, a 1 followed by 17 zeros, a number so large that it strains comprehension. Wolfenden's work, and the work of others who followed, revealed that arotidine decarboxylase was not unique in achieving such astronomical accelerations. Other enzymes showed similar enhancements, accelerating reactions by factors of 10 to the 15th, 10 to the 16th, 10 to the 19th. These were not marginal improvements, not the kind of modest rate enhancements that might be explained by simple transition state stabilization. These were accelerations so dramatic that they pushed against the boundaries of what transition state theory alone could comfortably explain. Scientists began to wonder whether something else might be going on, some additional factor or mechanism that was contributing to catalysis in ways that the classical models did not capture. It was around this time that researchers began to consider a possibility that had long been dismissed as implausible, the possibility that quantum mechanical effects might play a role in enzyme catalysis. Quantum mechanics, as you may know, is the branch of physics that describes the behavior of matter at very small scales, at the level of atoms and subatomic particles. It is a strange and counterintuitive theory, full of phenomena that seem impossible according to our everyday experience, such as particles passing through barriers they should not be able to cross, or existing in multiple states simultaneously until observed. For most of the 20th century, the conventional wisdom held that quantum effects were relevant only at very low temperatures and in carefully isolated systems, and that in the warm, wet, noisy environment of a living cell, any quantum coherence would be destroyed almost immediately by thermal fluctuations and interactions with surrounding molecules. Biology, it was thought, was simply too messy for quantum mechanics to matter. This skepticism was not unreasonable, really, for it was based on solid physical principles about how quantum systems interact with their environments. The phenomenon known as decoherence, whereby quantum superpositions are rapidly destroyed by environmental interactions, seemed to preclude any meaningful quantum effects in biological systems operating at physiological temperatures. Cells are warm, typically around 37 degrees Celsius in mammals, and they are filled with water molecules constantly jostling and colliding with everything around them. It seemed inconceivable that quantum coherence could survive long enough in such an environment to influence the outcome of a chemical reaction. And so for many years, when quantum effects were proposed as explanations for biological phenomena, the proposals were met with polite skepticism or outright dismissal. But science has a way of surprising us, of revealing depths where we expected shallows and complexity where we presumed simplicity. As experimental techniques grew more sophisticated and theoretical frameworks more nuanced, researchers began to accumulate evidence that perhaps the conventional wisdom was not entirely correct. Perhaps, in certain specific contexts, biological systems had found ways to exploit quantum mechanical effects despite the apparent hostility of their cellular environment. Perhaps enzymes, those ancient molecular machines refined by billions of years of evolution, had stumbled upon tricks that our theories had not anticipated. This possibility, once considered fanciful, has gradually grown more respectable over the past few decades, as experiments have revealed phenomena that seem difficult to explain without invoking quantum mechanics. And so we find ourselves at the threshold of a new understanding, one that does not discard the insights of Fisher or Pauling or Koshland or Michaelis and Mentén, but rather builds upon them, adding new layers to our comprehension of how enzymes achieve their remarkable feats of catalysis. The lock and key model still captures something essential about enzyme specificity, and transition state stabilization remains central to our understanding of catalysis, and induced fit dynamics continue to explain the flexibility and responsiveness of enzyme structure. But to these classical ideas we may need to add something more, something borrowed from the quantum world, something that operates at a level deeper than the motions of whole atoms, at the level of electrons and protons themselves. This is the story we shall continue to explore, gently and without haste, in the sections that follow, as we consider how the strange rules of quantum mechanics might be woven into the fabric of life itself, helping enzymes to work their quiet miracles in every cell of every living thing. Let us now turn our attention to a rather unassuming enzyme, one that by all classical measures should not function nearly as efficiently as it does, and yet operates with a quiet reliability that puzzled researchers for many years before a deeper understanding began to emerge. This enzyme, known as aromatic amine dehydrogenase, or simply ADH, became something of a landmark in the study of quantum effects in biology, and the story of how scientists came to appreciate its unusual behavior is a gentle one, unfolding over careful experiments and patient analysis in laboratories primarily at the University of Manchester, where a researcher named Nigel Scruton dedicated much of the early 2000s to understanding what made this particular enzyme so puzzling. Aromatic amine dehydrogenase is found in certain bacteria, where it performs the rather specialized task of processing aromatic amines, which are molecules containing ring structures attached to nitrogen-containing groups. The enzyme helps break down these compounds as part of the bacterium's metabolism, extracting electrons in a process that seems straightforward enough when described in broad terms. However, when Scruton and his colleagues began examining the fine details of how this reaction actually proceeds, they encountered measurements that simply did not fit within the framework of classical chemistry, and it was this mismatch between expectation and observation that would eventually lead to a much richer understanding of how biology can harness quantum mechanical phenomena. At the heart of aromatic amine dehydrogenase lies a most unusual cofactor, a small molecule that assists the enzyme in carrying out its chemical work. This cofactor is called tryptophan tryptophyllquinone, often abbreviated as TTQ, and it represents a rather remarkable piece of biological engineering. Unlike many cofactors that are synthesized separately and then inserted into enzymes, TTQ is actually formed from two amino acids that are already part of the enzyme's own protein chain. These two tryptophan residues become chemically modified and cross-linked to one another, creating a structure capable of accepting and donating electrons in ways that ordinary amino acids cannot. The resulting TTQ cofactor has a distinctive quin-1 group, a chemical arrangement involving oxygen atoms double bonded to a ring structure, and this quin-1 can cycle between different oxidation states as it participates in the enzyme's catalytic cycle. The chemistry of TTQ is inherently interesting, but what made aromatic amine dehydrogenase particularly compelling to researchers was the way it handles hydrogen atoms during catalysis. When the enzyme processes its substrate, it must transfer both protons and electrons, and it does so through a mechanism known as proton-coupled electron transfer. In this process, a proton, which is simply a hydrogen atom stripped of its electron, moves in concert with electron transfer, the two events intimately linked rather than occurring as separate sequential steps. This coupling presents particular challenges for classical chemical theory, because protons are relatively heavy particles compared to electrons, and moving them requires overcoming significant energy barriers that would typically slow reactions considerably. The puzzle deepened when Scruton's group began measuring something called kinetic isotope effects. In these experiments, researchers replace ordinary hydrogen atoms with deuterium, a heavier isotope of hydrogen that has an additional neutron in its nucleus. Because deuterium is heavier, it moves more slowly according to classical mechanics, and reactions involving deuterium typically proceed at a slower rate than those with ordinary hydrogen. By comparing reaction rates with hydrogen versus deuterium, scientists can learn a great deal about whether hydrogen transfer is the rate-limiting step and about the mechanism by which that transfer occurs. The kinetic isotope effects observed in aromatic amine dehydrogenase were substantial, indicating that hydrogen transfer was indeed central to the enzyme's function. But there was something else in the data that defied easy explanation. When the Manchester group examined how the kinetic isotope effect changed with temperature, they found something quite unexpected. In classical chemistry, kinetic isotope effects typically vary with temperature in predictable ways, becoming larger at lower temperatures where the energy available to climb over reaction barriers is reduced. However, in aromatic amine dehydrogenase, the kinetic isotope effect remained essentially constant across a remarkably wide temperature range, from about 5 degrees Celsius all the way up to 55 degrees Celsius. This temperature independence was deeply puzzling from a classical perspective, because it suggested that the hydrogen transfer mechanism was somehow insensitive to the thermal energy available in the system. Classical over-the-barrier processes simply do not behave this way, and the flat temperature dependence pointed toward a fundamentally different mechanism at work. The explanation that emerged, supported by careful experimental work and theoretical analysis, was that hydrogen transfer in aromatic amine dehydrogenase occurs primarily through quantum mechanical tunneling. Rather than climbing over the energy barrier that separates reactants from products, the hydrogen nuclei were passing through the barrier, exploiting the wave-like nature of matter at small scales. Quantum tunneling had been known to physicists for many decades, but its importance in enzyme catalysis was only beginning to be fully appreciated, and aromatic amine dehydrogenase provided some of the clearest evidence that this quantum phenomenon could be central to biological function. A particularly significant contribution came in 2006, when Scruton and colleagues published a paper in the journal Science that helped clarify how the enzyme achieves such efficient tunneling. The key insight was that the enzyme does not simply provide a passive environment in which tunneling might occasionally happen, but rather actively organizes its active site to make tunneling highly probable. This concept of pre-organization proved essential to understanding the temperature-independent kinetic isotope effects, because it suggested that the enzyme structure is already optimized to position the hydrogen donor and acceptor at precisely the right distance and orientation for tunneling to occur readily. The idea of promoting vibrations became central to this understanding. Enzymes are not rigid structures but rather dynamic assemblies that constantly flex and breathe, sampling different conformations on timescales ranging from femtoseconds to milliseconds. Scruton and others proposed that certain specific protein motions, rather than being incidental fluctuations, actually play a functional role in catalysis by transiently compressing the distance between the donor and acceptor groups involved in hydrogen transfer. When this distance is reduced, even by a small fraction of an angstrom, the probability of tunneling increases dramatically, because tunneling efficiency depends exponentially on barrier width. These promoting vibrations thus act as a kind of mechanical assistance, rhythmically creating windows of opportunity during which tunneling becomes highly favorable. The temperature independence of the kinetic isotope effect could now be understood in this framework. If the enzyme structure is already pre-organized to minimize the tunneling distance, and if the promoting vibrations that further compress this distance are relatively temperature insensitive, then the tunneling probability would remain roughly constant regardless of whether the system was cold or warm. The classical temperature dependence that one might expect simply does not apply, because the reaction proceeds not by thermally activated barrier crossing but by quantum mechanical penetration through a barrier that the enzyme has evolved to make as narrow as possible. This work on aromatic amine dehydrogenase did not occur in isolation but was enriched through collaboration with other leading figures in the field. Judith Klinman, whose pioneering studies at the University of California, Berkeley had helped establish the importance of hydrogen tunneling in enzymes years earlier, contributed her deep expertise in kinetic isotope effect methodology and interpretation. The collaboration between the Manchester Group and Klinman's laboratory strengthened the conclusions that could be drawn from the experimental data and helped situate the findings within the broader context of enzyme catalysis research. Computational work provided another essential perspective, helping to bridge the gap between experimental observations and theoretical understanding. Sharon Hammes-Schiffer, working initially at Pennsylvania State University and later at other institutions, developed sophisticated theoretical frameworks for describing proton-coupled electron transfer reactions in enzymes. Her calculations helped illuminate how the coupling between electronic and nuclear motion in aromatic amine dehydrogenase facilitates the overall catalytic process, and her models provided quantitative predictions that could be tested against experimental measurements. The interplay between experiment and theory proved particularly fruitful, with each informing and refining the other in an iterative process that gradually deepened understanding. As the research matured, aromatic amine dehydrogenase became recognized as a model system for studying quantum tunneling in biology. Its relatively simple active site architecture, combined with the clarity of the tunneling signatures in its kinetic behavior, made it an ideal subject for detailed investigation. Researchers could probe how mutations in the protein affected tunneling efficiency, how the dynamics of the protein scaffold influenced the reaction, and how the pre-organization of the active site was achieved at a molecular level. Each study added to a growing picture of an enzyme that has been refined by evolution to exploit quantum mechanical effects in a way that enhances its catalytic power beyond what classical chemistry would permit. The implications extended well beyond this single enzyme. If aromatic amine dehydrogenase had evolved to utilize tunneling, it seemed likely that other enzymes might have done the same, and indeed subsequent research has identified tunneling contributions in a wide variety of biological catalysts. The concepts developed through studying AADH, including the importance of pre-organization, the role of promoting vibrations, and the diagnostic value of temperature-independent kinetic isotope effects, became tools that researchers could apply to other systems. What had once seemed an anomaly, an enzyme that should not work as well as it does, became a window into a broader principle of biological chemistry. The story of aromatic amine dehydrogenase is in many ways a quiet one, lacking the drama of sudden discoveries or revolutionary upheavals. It unfolded through careful measurement and patient analysis, through the accumulation of evidence that gradually shifted understanding. Nigel Scruton and his colleagues, along with collaborators like Klinman and Hamas Schiffer, assembled a picture of enzyme function that integrated quantum mechanics into the fabric of biochemistry in a way that earlier generations of scientists might not have anticipated. The enzyme itself continues its modest work in bacterial cells, indifferent to the attention it has received, catalyzing its reactions with the same efficiency it always has. But for those who study it, aromatic amine dehydrogenase represents something rather profound, a reminder that the boundaries between classical and quantum worlds are not as sharp as one might suppose, and that biology, in its slow, patient way, has found means to harness phenomena that physicists once thought confined to atoms and subatomic particles. The TTQ cofactor cycles through its oxidation states, protons tunnel through barriers that they cannot classically surmount, and the enzyme continues to function, as it has for millions of years, guided by principles that researchers are still working to fully comprehend. It is a gentle sort of mystery, the kind that deepens rather than resolves with study, and it speaks to the remarkable subtlety of the molecular machinery that sustains all living things. And now we come to something rather wonderful, a story about birds and their long journeys across the earth, and about how they might find their way using abilities that seemed, for many years, too strange for biology textbooks to accept. This is a gentle sort of story, one that unfolds across decades and across continents, involving migratory birds and the patient work of scientists who suspected that navigation in the natural world might depend upon the most delicate quantum mechanical processes imaginable. Consider, if you will, the European robin, that small russet-breasted bird known to ornithologists as Erythacus rubecula, which each autumn departs from its breeding grounds in Scandinavia and travels southward across the vastness of Europe, crossing mountain ranges and coastlines and agricultural plains, until it arrives at its wintering grounds along the warm shores of the Mediterranean Sea. These journeys span thousands of kilometers, and the birds undertake them with remarkable precision, returning year after year to the same general locations, navigating through darkness and through clouded skies when neither sun nor stars are visible to guide them. For centuries, naturalists wondered how such small creatures accomplished these feats of orientation, and while it was understood that birds could sense the magnetic field of the Earth, the mechanism by which they did so remained elusive, wrapped in mystery and speculation. The story of how we came to understand this mechanism begins in 1978, at the Max Planck Institute in Germany, where a theoretical physicist named Klaus Schulten proposed something that his colleagues found rather difficult to believe. Schulten was interested in how chemical reactions might be influenced by magnetic fields, and he had been studying a particular type of chemistry involving what are called radical pairs, which are pairs of molecules that each contain an unpaired electron, and which are formed together in such a way that the quantum mechanical spins of those two unpaired electrons become correlated, or entangled, at the moment of their creation. Now, an unpaired electron is a somewhat restless thing, chemically speaking, and the spin of an electron is a quantum property that determines how it interacts with magnetic fields, and Schulten realized that the entangled spins of a radical pair would be exquisitely sensitive to very weak magnetic fields, including fields as weak as the Earth's magnetic field, which is roughly 50 microtesla in strength, about 100 times weaker than a small refrigerator magnet. What Schulten proposed was that this sensitivity might form the basis of a biological compass, a way for living organisms to detect the direction of the Earth's magnetic field using nothing more than the quantum behavior of electron spins in carefully arranged molecules. At the time, this hypothesis was met with considerable skepticism, because the prevailing view was that quantum effects were far too fragile to survive in the warm, wet, noisy environment of a living cell. Quantum coherence, the physicists said, required isolation from the environment, and biology was about as far from isolated as one could imagine. But Schulten persisted, and over the following decades, evidence began to accumulate that his radical pair mechanism might indeed be operating inside the bodies of migratory birds. The key discovery came when researchers identified a family of proteins called cryptochromes, which are found in many organisms and which are known to be sensitive to blue light. Cryptochromes were first studied in plants, where they play a role in regulating growth patterns in response to light, but they were subsequently found in animals as well, including in the retinas of birds. And here is where the story becomes particularly interesting, because the retina is where vision happens, and the presence of cryptochromes in the eyes of migratory birds suggested that these creatures might not merely sense the magnetic field but might actually see it, perceiving magnetic information as a kind of visual pattern overlaid upon their ordinary view of the world. In the European robin, a specific cryptochrome called CRY4 has been the subject of intensive study. This protein is expressed at high levels in the outer segments of the photoreceptor cells in the bird's retina, exactly where you would expect to find a molecule involved in gathering information from the environment. When blue light strikes the cryptochrome molecule, it triggers a series of chemical reactions that result in the transfer of an electron from one part of the molecule to another, creating a pair of radicals whose electron spins are quantum mechanically entangled. This radical pair exists in a delicate superposition of quantum states, and the particular mixture of states shifts depending on the orientation of the molecule relative to the Earth's magnetic field. Different orientations lead to different chemical outcomes, and these differences can in principle be detected by the bird's nervous system, providing information about which way is north. Now, you might reasonably wonder how scientists could test such a hypothesis, and the answer involves a series of elegant experiments conducted over many years. Some of the most important early work was carried out in 2008 by a research group led by Thorsten Ritz at the University of California, Irvine. Ritz and his colleagues designed experiments in which migratory birds were placed in orientation cages, which are funnel-shaped enclosures that allow researchers to record which direction a bird is trying to fly based on the marks its movements leave on the walls of the funnel. Under normal conditions, the robins oriented themselves correctly, pointing in the direction of their intended migration. But when the researchers applied weak radiofrequency electromagnetic fields oscillating at specific frequencies, the birds became disoriented, unable to determine which way to go. This finding was significant because the frequencies that disrupted the bird's navigation were precisely the frequencies predicted to interfere with the radical pair mechanism. You see, the entangled electron spins in a radical pair process, or wobble, at rates determined by their magnetic environment, and oscillating fields at certain frequencies can disrupt the delicate quantum dance of these spins, scrambling the information they carry. The fact that robins lost their navigational ability when exposed to these specific radio frequencies provided strong support for Schulten's hypothesis, suggesting that the birds were indeed relying on quantum coherent processes to sense the magnetic field. Other explanations for magnetic sensing, such as mechanisms involving tiny crystals of magnetite, would not have been affected by these particular radio frequencies, and so the experiments helped to distinguish between competing theories. Further evidence for the quantum compass came from work conducted by Henrik Moritzen and his colleagues at the University of Oldenburg in Germany. Moritzen's group investigated the neural pathways involved in magnetic sensing, searching for the brain regions that process magnetic information. They discovered that a specific cluster of neurons, which they named Cluster N, located in a part of the bird's brain associated with visual processing, showed high levels of activity when the birds were engaged in magnetic orientation behavior. When Cluster N was surgically lesioned, the birds lost their ability to use the magnetic compass, though they could still navigate using the sun and the stars when these were visible. This finding reinforced the idea that magnetic information was being processed through the visual system, consistent with the hypothesis that the birds were literally seeing the magnetic field. And what might that experience be like, to see a magnetic field? Scientists have speculated, based on mathematical models of how cryptochrome signals might be processed by the visual system, that birds might perceive magnetic field lines as patterns of light or shadow, perhaps as a kind of filtering or shading effect that varies depending on which direction the bird is looking. Imagine, if you will, a subtle gradient across the visual field, darker in one direction and lighter in another, that shifts as the bird turns its head. This pattern would not obscure the bird's ordinary vision but would be layered atop it, providing a constant reference for direction. It is a gentle sort of augmented reality, if you like, woven into the fabric of everyday perception, and while we cannot know with certainty what the bird experiences, the neurological evidence suggests that magnetic information and visual information are processed together as aspects of a single integrated perception of the world. The quantum mechanical details of how this all works are subtle but need not be alarming. When blue light strikes the cryptochrome protein, an electron is excited and transferred from a molecule called flavin adenine dinucleotide, which is bound within the protein, to a series of tryptophan amino acid residues arranged in a chain. This transfer creates the radical pair, with one unpaired electron on the flavin and another on a tryptophan. The spins of these two electrons, having been created together in a single quantum event, are entangled in what is called a singlet state, meaning their spins are correlated in a particular way. But as time passes, the spins evolve under the influence of local magnetic fields, including the Earth's field, and this evolution causes the radical pair to oscillate between the singlet state and a triplet state, where the spins are correlated differently. The rate and pattern of this oscillation depend on the orientation of the cryptochrome molecule relative to the magnetic field, and because singlet and triplet pairs undergo different chemical reactions, the outcome of the process, which chemical products are formed, varies with orientation. In 2021, a landmark study demonstrated that cryptochrome proteins from the European robin are indeed sensitive to magnetic fields when tested in the laboratory, outside the bird's body entirely. Researchers at the University of Oxford, working with collaborators in Germany, purified CRY4 protein and showed that its photochemistry was influenced by magnetic fields of similar strength to the Earth's. This was an important confirmation, because it demonstrated that the radical pair mechanism was not merely a theoretical possibility but an actual property of the specific molecules found in the bird's eyes. The study used sophisticated spectroscopic techniques to observe the radical pairs in action, watching as their quantum states evolved and as the magnetic field altered the balance of chemical products. It is worth pausing here to appreciate how remarkable this is. The Earth's magnetic field is very weak, far weaker than the magnetic fields used in medical imaging or industrial applications, and yet these small proteins in the eyes of songbirds can detect it, translating quantum mechanical phenomena into signals that the nervous system can interpret. The process depends on quantum coherence, on the delicate entanglement of electron spins, and on the fact that this coherence can persist long enough, perhaps for microseconds or tens of microseconds, for the magnetic field to have its effect. The warm, wet environment of the living eye does not immediately destroy the quantum information, because the cryptochrome protein provides a degree of protection, holding the radical pair in a sheltered configuration where the most destructive types of environmental noise are suppressed. And so, each autumn, when European robins depart from their northern breeding grounds and set their course toward the Mediterranean, they carry with them these exquisite quantum sensors, inherited from countless generations of ancestors who made the same journey. The birds fly through the night, when the air is calm and predators are fewer, and as they fly they see the magnetic field stretching before them, a faint pattern of light and shade that tells them which way is south. They navigate not by conscious calculation but by following what appears to them as simply another feature of the visible world, as natural and immediate as the shape of the horizon or the brightness of the stars. Klaus Schulten, who first proposed this mechanism in 1978, passed away in 2016, but not before seeing his once controversial hypothesis vindicated by decades of experimental work. The quantum compass of migratory birds stands today as one of the clearest examples of quantum biology, a field that continues to reveal the ways in which life has learned to exploit the strange rules of the quantum world. And on quiet autumn evenings, as robins lift into the darkening sky and begin their long journeys south, the physics of electron spins and radical pairs goes with them, guiding them faithfully across the turning earth, a compass woven into the very act of seeing, as gentle and constant as the light. And so we come now to a rather intriguing corner of quantum biology, one that involves something you do thousands of times each day without the slightest conscious thought, which is of course the act of breathing in the world around you and somehow knowing, in that immediate and wordless way, that you are smelling coffee or rain on warm pavement or the particular green scent of crushed leaves. This is the sense of smell, known more formally as olfaction, and it has puzzled scientists for a very long time because it seems almost impossibly precise and yet operates through mechanisms that remain, even now, not entirely settled. The story we are about to explore involves a biophysicist with an unusual second career as a perfume critic, a theory that most experts initially dismissed, some rather elegant experiments involving flies and heavy hydrogen, and the kind of ongoing scientific disagreement that unfolds slowly over decades, which is precisely the sort of gentle controversy that makes for restful contemplation as you settle in for the evening. The man at the center of this particular scientific puzzle is Luca Turin, who was born in Lebanon to an Italian diplomat father and spent portions of his childhood in Paris, which may or may not explain his later fascination with the art and science of perfumery. Turin trained as a biophysicist and worked for many years in research positions at various institutions, but he also developed what can only be described as an extraordinarily refined appreciation for perfumes, eventually authoring a book called Perfumes: The Guide alongside the writer Tania Sanchez. This book offered ratings and reviews of hundreds of commercial fragrances, written with the kind of precise descriptive language usually reserved for wine criticism or literary analysis, and it established Turin as a figure who moved between the worlds of hard science and aesthetic appreciation with unusual fluency. This dual identity would prove important when Turin proposed, in 1996, a theory of how the nose actually works that departed significantly from the accepted understanding, because his intimate familiarity with real perfumes and their subtle distinctions gave him an intuition about olfaction that pure laboratory scientists might not have possessed. For most of the 20th century, the dominant explanation for how we smell things was what scientists call the shape theory, or sometimes the lock and key model, which proposes that odor molecules are detected because of their three-dimensional shapes. In this view, the olfactory receptors in your nose, of which humans possess somewhere around 400 different types, are like locks waiting for specifically shaped keys, and when a molecule of the right shape drifts in and fits snugly into a receptor, it triggers a signal that travels to your brain and is interpreted as a particular smell. This is a satisfyingly mechanical picture, rather like puzzle pieces clicking together, and it explained a great deal about why differently shaped molecules tend to smell different from one another. The shape theory received powerful support in 1991 when Linda Buck and Richard Axel published their Nobel Prize-winning work describing the large family of olfactory receptor genes, demonstrating that the nose uses a combinatorial code of receptor activations to distinguish among the thousands of different odors that humans can perceive. Shape theory seemed to be working well enough, and most researchers were content to refine the details rather than seek alternative explanations. But Turin had noticed certain things that did not fit neatly into the shape theory, observations that accumulated gradually over his years of thinking about perfumes and molecules. He noticed, for instance, that molecules with quite different shapes sometimes smell remarkably similar to one another, while molecules with nearly identical shapes sometimes smell quite different. The relationship between molecular structure and perceived odor was messier than a simple lock-and-key model would predict. There was also the curious observation that some molecules containing sulfur smell distinctly sulfurous, that characteristic unpleasant note found in rotten eggs and cooked cabbage, even when those molecules have different shapes, as though the nose were somehow detecting the presence of sulfur itself rather than just the overall molecular geometry. Turin began to wonder whether the nose might be sensing something else entirely, some property of molecules that cut across their various shapes, and he eventually arrived at an old and largely forgotten idea from the 1930s, which proposed that the nose might detect the vibrational frequencies of molecular bonds. Every molecule vibrates, you see, and the bonds between atoms within a molecule oscillate at specific frequencies determined by the masses of the atoms involved and the strength of the bonds connecting them. These vibrations occur in the infrared range of the electromagnetic spectrum, and they can be measured using a technique called infrared spectroscopy, which has been a standard tool of chemistry for many decades. Different molecules have different vibrational spectra, like fingerprints, and Turin proposed that olfactory receptors might somehow be detecting these vibrations rather than, or perhaps in addition to, molecular shapes. The mechanism he suggested was inelastic electron tunneling, a quantum mechanical process in which an electron tunnels through a potential barrier but loses a precise amount of energy in doing so, energy that corresponds to the vibrational frequency of a molecular bond. In this view, when an odor molecule sits within an olfactory receptor, an electron attempts to tunnel from one side of the receptor to the other, and the success of this tunneling depends on whether the molecule can absorb the energy difference by vibrating at the right frequency. If the frequencies match, the electron tunnels, a signal is generated, and you perceive a smell. Turin published this theory in the journal Chemical Senses in 1996, and the reception from the mainstream scientific community was, to put it gently, skeptical. The primary objection was that no one had ever demonstrated electron tunneling in biological olfactory receptors, and the idea seemed to require a degree of molecular precision and quantum coherence that many scientists doubted could exist in the warm, wet, noisy environment of a working nose. The olfactory system is not like a carefully controlled physics laboratory, critics pointed out, and maintaining quantum effects at body temperature seemed implausible. There were also questions about the biophysics, about whether the receptor proteins could be structured in a way that would allow the proposed tunneling mechanism to function. Turin's theory remained on the fringes of olfactory science for some years, a curiosity that most researchers chose to ignore rather than actively investigate. But Turin and his collaborators continued to pursue experimental tests of the vibrational theory, and one particularly elegant approach involved isotopes, which are atoms of the same element that differ only in the number of neutrons they contain. Hydrogen, the simplest element, can exist as normal hydrogen with no neutrons, or as deuterium with one neutron, or as tritium with two neutrons. When you replace the hydrogen atoms in a molecule with deuterium atoms, you create a deuterated version of that molecule that has exactly the same shape as the original, the same size, the same distribution of electrical charge, but different vibrational frequencies, because the heavier deuterium atoms vibrate more slowly than normal hydrogen. If the nose detects only shape, then normal and deuterated versions of the same molecule should smell identical. But if the nose detects vibrations, they might smell different, because their vibrational spectra would be shifted to lower frequencies. Turin and collaborators at the Massachusetts Institute of Technology conducted experiments using the common fruit fly, Drosophila melanogaster, which is a favorite organism of biologists because of its simple genetics and well-understood nervous system. They trained flies to associate certain odors with rewards or punishments and then tested whether the flies could distinguish between normal and deuterated versions of the same molecules. The results were striking, in a quiet scientific way, because the flies appeared able to tell the difference between isotopic versions that were identical in shape but different in vibration. Similar experiments were conducted with humans, using molecules such as acetophenone, which is a simple aromatic compound used in flavorings and perfumes. When human subjects were asked to compare normal acetophenone with deuterated acetophenone, some studies found that people could indeed distinguish between them, perceiving subtle differences in odor quality despite the molecules having identical shapes. The vibrational frequencies that Turin proposed were relevant fell in the range of roughly 1500 to 3500 reciprocal centimeters, which corresponds to the stretching vibrations of carbon-hydrogen bonds, a region where deuteration produces noticeable shifts. These results were intriguing but not definitive, and the broader scientific community remained divided. In 2011, a paper appeared in the Proceedings of the National Academy of Sciences that attempted to replicate some of the isotope discrimination findings and to test predictions of the vibrational theory more rigorously. The results were mixed, with some findings supporting vibrational detection and others being more ambiguous, and the paper sparked further debate rather than settling the question. Then, in 2015, a carefully designed study from the laboratory of Leslie Vosshall at Rockefeller University was published, and it reported negative results. Vosshall and her colleagues tested whether humans could discriminate between deuterated and non-deuterated musks, molecules specifically chosen because they were predicted by Turin's theory to show clear vibrational differences that should be perceptible. The study found that subjects could not reliably distinguish between the isotopic variants, which appeared to contradict the vibrational theory, and Vosshall and her co-authors concluded that their results did not support the idea that humans smell molecular vibrations. This is where the story remains, in a state of gentle scientific uncertainty that may persist for years or even decades to come. Some researchers believe that the negative results from the Vosshall lab effectively disprove the vibrational theory, at least as a general mechanism of olfaction, while Turin and his supporters argue that the particular molecules tested were not ideal and that the theory remains viable for other classes of odors. It is worth noting that science often proceeds through exactly this kind of protracted disagreement, with different laboratories obtaining different results and interpretations shifting slowly as evidence accumulates. The question of how we smell remains more open than textbooks sometimes suggest, and the possibility that quantum mechanical effects play some role in olfaction has not been entirely ruled out, even if it has not been definitively established either. What is perhaps most interesting, as you lie there drifting towards sleep, is not the resolution of this particular debate but the fact that the debate exists at all, because it reminds us that the boundary between classical and quantum behavior in biology is genuinely uncertain and may harbor surprises. The human nose, that humble protuberance in the middle of your face, might be a quantum measurement device of exquisite sensitivity, or it might simply be a very sophisticated shape detector, or it might be some combination of both that we do not yet have the concepts to describe. Turin himself continues to work on the problem, bringing to it his unusual combination of biophysical training and perfumer's intuition, and other researchers continue to design experiments that might distinguish more clearly between the competing theories. The vibrations of molecular bonds continue to occur whether we can smell them or not. Countless trillions of tiny oscillations in the air around you as you breathe in and out, and somewhere in that ceaseless molecular dance there may be secrets about olfaction that remain to be discovered. For now, you need not resolve this question, and indeed resolution is not the purpose of our time together. The uncertainty itself is restful, in a way, because it reminds us that the world is larger and stranger than any single theory can capture, and that the patient accumulation of evidence, experiment by experiment and year by year, is itself a kind of meditation on the nature of things. Luca Turin continues to write about perfumes with the same precision he brings to physics, describing the ineffable beauty of complex odors in language that attempts to bridge the gap between sensation and understanding, and in his own way he embodies the deep mystery of olfaction, which is how something as simple as a molecule drifting into your nose can evoke memories and emotions and subtle aesthetic judgments that seem far removed from the cold equations of chemistry and physics. Whether the nose is a quantum device or a classical one, it remains a small miracle of biology, and the controversy surrounding its operation is one more gentle reminder that the sciences have not exhausted their capacity to surprise us, even in the most familiar aspects of our everyday experience. Let us now turn our attention to something rather extraordinary, though we shall approach it with the same gentle curiosity we have maintained throughout our time together this evening. We are going to consider how the quantum mechanical phenomena we have been discussing, these strange tunneling events that allow particles to pass through barriers they ought not to pass through, might play a role in the very fabric of life itself, in the long spiraling molecule that carries the instructions for building and maintaining every living thing on Earth. This is the story of how protons, those smallest of atomic nuclei, might occasionally find their way to places they should not be, and in doing so, introduce small changes into the genetic code, changes that we call mutations, and how this possibility was first seriously proposed by a Swedish scientist working in the university town of Uppsala more than 60 years ago. The year was 1963, and Per-Olof Lodin, a theoretical chemist of considerable reputation, was thinking about the double helix structure of DNA that had been described by Watson and Crick just a decade earlier. Lodin was interested in something that might seem at first to be a minor chemical detail, but which he suspected could have profound implications for biology. He was thinking about the hydrogen atoms that hold the two strands of the double helix together, and specifically about the protons, the positively charged nuclei of those hydrogen atoms, and whether they might sometimes behave in ways that classical chemistry would not predict. Working at Uppsala University in Sweden, where he had established a renowned research group in quantum chemistry, Lodin proposed that the protons involved in the hydrogen bonds between DNA base pairs might occasionally tunnel from one position to another, and that when this happened, it could lead to changes in the genetic code that would be passed on when the DNA was copied. This was a remarkable suggestion, linking the abstract mathematics of quantum mechanics to the biological reality of heredity and mutation. To understand what Lodin was proposing, we need to consider how the base pairs in DNA are normally arranged. The two strands of the double helix are held together by hydrogen bonds between complementary bases, adenine pairing with thymine, and guanine pairing with cytosine. In each of these pairings, hydrogen atoms are shared in a very specific way, with the protons sitting at particular locations along the hydrogen bonds. In the normal arrangement of adenine and thymine, for instance, there are two hydrogen bonds connecting them, and the protons in these bonds occupy positions that we might call their canonical or standard locations. This standard arrangement is what allows DNA replication to work reliably, because when the double helix is unzipped and each strand serves as a template for building a new complementary strand, the replication machinery recognizes each base by its shape and its pattern of hydrogen bond donors and acceptors, and adds the correct complementary base accordingly. But here is where things become interesting in a quiet sort of way. The bases in DNA can exist in alternative forms called tautomers, which differ from the standard forms in the positions of certain protons. Adenine, for example, normally exists in what chemists call its amino form, but it can also exist in a rare imino form, where a proton has moved from one position to another on the molecule. Similarly, thymine normally exists in its chato form, but can shift to a rare enol form through a similar proton rearrangement. These alternative tautomeric forms are called rare because under normal circumstances, the standard forms are much more stable, and the vast majority of bases at any given moment exist in their canonical configurations. The shift from one tautomeric form to another involves a proton moving across a small energy barrier, and classically, this would require the proton to have enough thermal energy to climb over that barrier. What Lodin proposed was that protons might not need to climb over the barrier at all. Instead, they might tunnel through it, in exactly the way we have been discussing throughout this narration. The quantum mechanical wave function of a proton extends through space, and there is always some probability, however small, that the proton will be found on the other side of an energy barrier, even if it never had enough energy to pass over the top. Lodin suggested that this tunneling could cause tautomeric shifts in DNA bases while they were still paired in the double helix, and that if such a shift occurred at just the wrong moment, during DNA replication, it could cause the replication machinery to add the wrong complementary base, resulting in a mutation that would be passed on to daughter cells. The mechanism works something like this, and we can trace it through slowly. Imagine an adenine-thymine base pair sitting quietly in the double helix. In its normal state, the adenine is in its amino form and the thymine is in its chato form, and they are paired correctly. Now suppose that while they are paired, a proton tunneling event occurs, causing the adenine to shift to its amino form and the thymine to shift to its enol form. This is called a double proton transfer, and it creates what we might call a tautomeric mispair. The shapes and hydrogen bonding patterns of these tautomeric forms are different from the standard forms, and here is the crucial point. The amino form of adenine has a hydrogen bonding pattern that resembles the standard form of guanine, while the enol form of thymine has a pattern that resembles cytosine. If the DNA is replicated while these tautomeric forms persist, the replication machinery may add the wrong complementary base, cytosine instead of adenine opposite the enol thymine, or thymine instead of cytosine opposite the aminoadenine. When this mismatched DNA is replicated again, a permanent mutation has been introduced into the genetic code. For many years, Lodin's hypothesis remained somewhat speculative, because it was difficult to measure whether proton tunneling in DNA actually occurred on timescales that were biologically relevant. The energy barriers involved are quite small, approximately 0.4 to 0.5 electron volts, which corresponds to a barrier that is low enough to make tunneling possible but high enough that the tunneling probability is not trivially large. The question was whether tunneling events occurred frequently enough, and whether the resulting tautomeric states persisted long enough to cause mutations at a rate that could be detected or that would matter biologically. This is where more recent work has begun to provide some answers. In 2022, a research team from the University of Surrey published a study that used sophisticated computational quantum mechanics to model proton tunneling in DNA base pairs. The researchers, led by physicists working in collaboration with biologists, applied techniques that could account for quantum effects with a precision that earlier calculations had lacked. Their computational models allowed them to simulate the behavior of protons in hydrogen bonds over time, accounting for the quantum mechanical uncertainties in proton position and the probability of tunneling through the energy barriers between tautomeric states. What they found was that tunneling does indeed occur on timescales that matter for biology. The calculations showed that proton tunneling events in guanine-cytosine base pairs occur at rates that produce tautomeric states lasting long enough to potentially cause mispairing during replication. The energy barriers of approximately 0.4 to 0.5 electron volts that we mentioned correspond to tunneling times that place these events within a range that is biologically significant, not so fast that the tautomers would convert back before replication could occur, and not so slow that they would be completely negligible. The Surrey team estimated that tautomeric states could persist for timescales comparable to or longer than the time required for a replication fork to pass a given base pair, meaning that the window for a mutation-causing mispair is real and not merely theoretical. To put this in context, let us consider the scale of the human genome. Human cells contain approximately 3 billion base pairs, and each time a cell divides, all of this DNA must be copied. If we estimate, based on these quantum mechanical calculations and related biochemical data, that perhaps 10 to 100 tautomeric events occur per cell division that have the potential to cause mutations, we begin to see how proton tunneling might contribute to the baseline rate of spontaneous mutations that are observed in living organisms. This is a small number compared to the total number of base pairs, which is why replication is generally very accurate. But over many cell divisions across billions of cells in a human body over decades of life, even small probabilities accumulate. Some fraction of the mutations that arise in our cells and that cannot be attributed to external causes, to ultraviolet radiation from the sun, or to chemical mutagens in our environment, or to errors in the replication machinery itself, might be traced to these quiet quantum mechanical events, protons finding their way to places they should not be through the peculiar physics of tunneling. The implications of this understanding extend in several directions, and we can consider them one at a time without any sense of urgency. For cancer research, the idea that proton tunneling contributes to spontaneous mutations is relevant because cancer arises from the accumulation of mutations in cells that control growth and division. Understanding all the sources of mutation, including those that are intrinsic to the quantum mechanical nature of the molecules themselves, helps researchers build more complete models of cancer risk and progression. It suggests that even in the complete absence of external mutagens, there is an irreducible flaw to the mutation rate, a quantum mechanical noise in the system that cannot be eliminated by any conceivable environmental or lifestyle intervention. This is not a cause for alarm, merely a recognition of the physical nature of the molecules that carry our genetic information. For evolutionary theory, the implications are similarly interesting in their quiet way. Evolution depends on mutation to generate the variation upon which natural selection acts. If some fraction of that variation arises from quantum mechanical processes rather than from classical chemical reactions or environmental damage, then in a sense quantum mechanics is one of the engines of evolutionary change. This does not alter the basic Darwinian picture. Mutations still arise, they are still subject to selection, and adaptation still occurs through the differential survival and reproduction of organisms with different genetic variants. But it adds a layer to our understanding of where those mutations come from in the first place, and it connects the physics of subatomic particles to the grand sweep of biological history in a way that Lodin, back in 1963, could only dimly anticipate. It is worth pausing to appreciate the intellectual journey that connects a theoretical proposal made in a Swedish university town in the early 1960s to the sophisticated computational quantum mechanics being performed in English laboratories 6 decades later. Lodin could not run the simulations that the Surrey team performed. He did not have access to computers capable of modeling the quantum behavior of protons in realistic molecular environments over biologically relevant timescales. He had intuition and mathematical reasoning, and he applied them to propose something that seemed possible based on the physics he understood, and then he waited to see if the tools would eventually exist to test his ideas. This is often how science works, with hypotheses running ahead of the technology needed to evaluate them, sometimes by years, sometimes by decades. The patience required is considerable, and the confirmation, when it comes, often arrives long after the original proposer has moved on to other questions or other pursuits. And so we find ourselves at a gentle resting point in this section, having traced the path from quantum mechanical tunneling, through the chemistry of DNA base pairs, to the biological reality of mutations and their consequences for health and evolution. The protons in our DNA are not classical particles sitting still in fixed positions. They are quantum mechanical objects with wave functions that extend through space, objects that can tunnel through barriers and find themselves in places that change the meaning of the genetic code. This happens rarely, perhaps a few tens of times each time a cell divides, but it happens, and when it does, it writes small changes into the instructions for life, changes that may be silent, or harmful, or occasionally, over long stretches of evolutionary time, beneficial. The quantum mechanics of proton tunneling, discovered in the abstract mathematics of wave equations, reaches down into the most intimate chemistry of living things and participates in the ongoing story of life on Earth. This is not dramatic, merely true, and it is a fine thing to carry with you as your eyes grow heavy and your thoughts begin to drift toward sleep. And now we come to the final destination of the cellular energy machinery, to the place where the electrons that have been passed along from one protein complex to another finally reach their ultimate purpose, which is to combine with oxygen and allow us to breathe, to live, to sustain the quiet miracle of existence that happens in every cell of your body as you lie there, settling into the comfortable darkness of the evening. This is the story of cytochrome C oxidase, which is also known as complex 4 in the electron transport chain, and it is perhaps the most remarkable enzyme in all of biology, not because it does anything particularly flashy or dramatic, but because it does something profoundly essential with an efficiency that borders on perfection, and it does so by harnessing the strange rules of quantum mechanics in ways that scientists are still working to fully understand, even now, decades after the first discoveries were made. Let us begin by appreciating just how important this enzyme truly is, in the quiet and understated way that important things often are. Cytochrome C oxidase is responsible for consuming approximately 90% of all the oxygen that you breathe into your lungs, which means that with every breath you take, the vast majority of those oxygen molecules are destined to meet their end in the depths of this particular protein complex, tucked away in the inner membranes of your mitochondria. The oxygen does not simply dissolve away or get used up in some vague and general sense, but rather it is specifically and deliberately consumed by this one enzyme, which takes the oxygen molecules and combines them with electrons and protons to form water, releasing energy in the process that your cells use to power everything they do. This might seem like a simple enough reaction when described in such plain terms, but the reality of how it happens is anything but simple, and it involves electrons traveling distances that should, according to the classical rules of chemistry, be impossible for them to traverse. The story of how we came to understand this process begins in the 1980s, at the California Institute of Technology, where a researcher named Harry Gray began investigating how electrons move through proteins. Gray was interested in a fundamental question that had puzzled scientists for years, which was how electrons could possibly travel the long distances required to get from one reactive site to another within these large biological molecules. In ordinary chemistry, electrons move through bonds, hopping from atom to atom in a predictable and well-understood fashion, but the distances involved in protein electron transfer were often far too great for this kind of direct movement, spanning 10 or even 14 angstroms across stretches of protein that contained no obvious conductive pathway. An angstrom, for those who find comfort in precise definitions, is 1/10,000,000th of a meter, which is roughly the diameter of a single hydrogen atom, and so 14 angstroms represents a distance that is still extraordinarily small by everyday standards but is nonetheless vast on the molecular scale, far greater than the length of any chemical bond. Gray and his colleagues discovered, through a series of careful experiments using ruthenium-modified proteins, that electrons were indeed capable of traversing these seemingly impossible distances, and they were doing so through a process called quantum mechanical tunneling. This is the same phenomenon we have discussed elsewhere in our gentle journey through the quantum mechanics of biology, wherein a particle such as an electron can pass through a barrier that classical physics would declare impassable, appearing on the other side without ever having occupied the space in between. The electron does not climb over the barrier or find a path around it, but rather it tunnels directly through, exploiting the fundamental uncertainty in its position that quantum mechanics allows. What Gray demonstrated was that this tunneling could occur over remarkably long distances in biological systems, far longer than anyone had previously thought possible, and this discovery opened up an entirely new field of research into how biology harnesses quantum effects for essential processes. Building upon Gray's experimental work, theoretical physicists David Berretan and Jose Anuchik developed what came to be known as the tunneling pathway model, which provided a framework for understanding how electrons find their way through the complex three-dimensional structure of proteins. According to this model, electrons do not simply tunnel through empty space in a straight line from one metal center to another, but rather they follow specific pathways through the protein medium, taking advantage of the covalent bonds, hydrogen bonds, and through space jumps that the protein structure provides. The protein, in this view, is not merely a passive scaffold holding the reactive sites in place, but rather an active participant in the electron transfer process, with its amino acid residues and its intricate folding pattern creating a kind of molecular circuitry that guides the tunneling electrons along favorable routes. Some pathways through the protein are better than others, conducting electrons more efficiently depending on how the atoms are arranged and how the bonds are oriented, and the enzyme has evolved over billions of years to optimize these pathways for maximum efficiency. Now let us turn our attention to the specific architecture of cytochrome C oxidase itself, to the metal centers that serve as the stations along this quantum highway, and to the remarkable choreography of electron transfer that allows this enzyme to do its work. The enzyme contains 4 metal centers that work together in careful coordination, and their names are Quor, Heme, Heme3, and Cub, each of which plays a specific role in the overall process. The Quor center is a binuclear copper site, meaning it contains 2 copper atoms held close together by the surrounding protein structure, and this is where electrons first enter the enzyme after being delivered by a small carrier protein called cytochrome C. The Quor center is located near the outer surface of the protein, positioned to accept electrons from cytochrome C and pass them along to the next station in the chain. From Quor, the electrons tunnel approximately 19 angstroms through the protein medium to reach heme A, which is an iron-containing porphyrin group similar to the heme found in hemoglobin. Heme serves as an intermediate way station, accepting electrons from Quor and then passing them along to the active site where the actual chemistry of oxygen reduction occurs. This intermediate step might seem unnecessary at first glance, an extra complication in what could otherwise be a more direct process, but it serves important functions in controlling the timing and energetics of the electron transfer, ensuring that everything happens in the proper sequence. The active site of the enzyme, where oxygen is actually consumed, consists of heme 3 and Cub working together as a binuclear center, with the ion of the heme and the copper positioned close enough to each other to jointly coordinate the oxygen molecule that will be reduced. When an oxygen molecule enters this active site, it binds to both metals simultaneously, and then through a carefully orchestrated series of electron and proton transfers, it is converted into water. The distances involved in these electron transfers are quite substantial by molecular standards. Electrons must tunnel approximately 13 angstroms from heme A to the heme 3 Cub active site, and the total distance from the initial electron donor to the final acceptor spans several tens of angstroms through the protein structure. Yet despite these distances, the electron transfer occurs with remarkable speed and efficiency, with nearly 100% of electrons successfully making the journey and contributing to the reduction of oxygen. This efficiency is truly extraordinary when one considers the challenges involved. The electron must find its way through a complex three-dimensional maze of atoms and bonds, tunneling through regions where classical physics would predict it could not possibly go, and it must do so quickly enough to keep up with the demands of cellular respiration, which requires the consumption of billions of oxygen molecules every second in every cell of your body. But the story of cytochrome C oxidase does not end with electron transfer, for there is another crucial function that this enzyme performs, and it is this function that connects its activity to the broader process of energy production in cells. As the enzyme reduces oxygen to water, it also pumps protons across the inner mitochondrial membrane, creating an electrochemical gradient that drives the synthesis of ATP, which is the molecular currency that cells use to power their activities. To understand why this proton pumping matters, we must briefly consider the chemiosmotic theory developed by the British biochemist Peter Mitchell in 1961, a theory that was initially met with considerable skepticism from the scientific community but was eventually vindicated, earning Mitchell the Nobel Prize in Chemistry in 1978. Mitchell proposed that cells generate ATP not through direct chemical coupling, as most scientists at the time believed, but rather through the establishment of a proton gradient across a membrane, with the energy stored in this gradient then being used to drive the synthesis of ATP by a molecular turbine called ATP synthase. This was a radical idea at the time, suggesting that the production of cellular energy depended on the physics of proton gradients and membrane potentials rather than on the chemistry of high-energy intermediates, and it took many years and many experiments before the scientific community came to accept its validity. Today, the chemiosmotic theory is recognized as one of the foundational principles of bioenergetics, and cytochrome C oxidase plays a central role in establishing the proton gradient that the theory describes. For each molecule of oxygen that cytochrome C oxidase reduces, the enzyme pumps 4 protons across the membrane, from the inside of the mitochondrion to the space between the inner and outer membranes. This proton pumping is mechanically coupled to the electron transfer process, meaning that the movement of electrons through the enzyme is linked to the movement of protons in a way that captures some of the energy released by oxygen reduction and stores it in the form of an electrochemical gradient. The mechanism by which this coupling occurs is still not fully understood, despite decades of research, but it is clear that the quantum mechanical tunneling of electrons is intimately connected to the classical mechanical movement of protons, with the enzyme somehow converting one form of motion into another with remarkable efficiency. This is one of the many places where the quantum and classical worlds meet within biology, where the strange rules of quantum mechanics interface with the more familiar rules of classical physics to produce the everyday miracle of cellular respiration. In 2010, researchers reported evidence of quantum coherence in the copper centers of cytochrome C oxidase, suggesting that the quasite in particular might exhibit coordinated quantum behavior that enhances its ability to accept and transfer electrons. Quantum coherence, you may recall from our earlier discussions, refers to a state in which quantum systems maintain a specific phase relationship with each other, allowing them to behave in a coordinated fashion rather than independently. The observation of quantum coherence in the copper centers was significant because it suggested that the enzyme might be actively exploiting quantum effects to improve its performance, rather than simply tolerating them as an unavoidable consequence of operating at the molecular scale. If the copper atoms in core are indeed behaving coherently, then they might be able to accept electrons from cytochrome C more efficiently, or to transfer those electrons to heme more rapidly, than would be possible if they were behaving as independent classical particles. This is still an active area of research, with scientists continuing to investigate the extent and significance of quantum effects in cytochrome C oxidase and other respiratory enzymes, but the emerging picture is one of biology as a remarkably sophisticated quantum engineer, having evolved over billions of years to harness the peculiar properties of matter at the smallest scales. And so we come to the end of our exploration of this particular enzyme, this quiet machine that sits in the darkness of your mitochondria and consumes the oxygen that you breathe, converting it to water and capturing energy with an efficiency that approaches perfection. It does this work without drama or fanfare, without any awareness of its own importance, simply following the laws of physics and chemistry that govern its behavior, laws that include the strange and counterintuitive rules of quantum mechanics. As you lie there in the comfortable darkness, breathing slowly and steadily, know that this enzyme is working within you, that electrons are tunneling through protein matrices and protons are being pumped across membranes, and that all of this activity is sustaining your life in ways that are both mundane and miraculous. And so we come now to a rather fascinating corner of modern science, where researchers are no longer content merely to observe the quantum behavior of enzymes but have begun, with considerable patience and ingenuity, to build quantum machines of their own, drawing inspiration from the billions of years of evolution that produced nature's most efficient catalysts, and attempting, in their quiet laboratories filled with humming computers and delicate glassware, to create artificial enzymes that harness the strange and wonderful effects of quantum mechanics, tunneling and coherence and zero-point energy, all in service of chemistry that might otherwise be impossible, or at the very least impractically slow. This emerging field, which sits at the intersection of computational biology and quantum physics and synthetic chemistry, represents one of the more ambitious undertakings in contemporary science, for it requires not only a deep understanding of how natural enzymes work, down to the quantum level, but also the creative capacity to design entirely new proteins that have never existed before, proteins that fold into precisely the right shapes, with active sites tuned to exploit quantum effects in ways that we are only beginning to understand, and the technical skill to actually synthesize these designs and test whether they perform as predicted, which, as you might imagine, they often do not, at least not on the first attempt, or the second, or sometimes even the 100th. At the University of Washington, in Seattle, where the gray skies and gentle rain perhaps encourage the sort of patient, methodical work that protein design requires, a researcher named David Baker has spent decades developing the tools and techniques that make this kind of molecular architecture possible, and his laboratory has become something of a beacon for scientists interested in creating proteins from scratch, what they call de novo design, from the Latin meaning from the beginning, and his work has fundamentally changed what we believe to be achievable in the realm of synthetic biology. The centerpiece of Baker's approach is a software suite called Rosetta, which began its development in the late 1990s and has been refined and expanded ever since, growing into an enormously sophisticated platform that can predict how amino acid sequences will fold into three-dimensional structures, and conversely, can work backwards from a desired structure or function to suggest sequences that might achieve it, a kind of molecular inverse problem that requires enormous computational resources and clever algorithms to solve even approximately. Rosetta works by exploring the vast landscape of possible protein conformations, searching for arrangements that minimize the free energy of the system, which is to say, arrangements that are thermodynamically stable, and it does this through a combination of physics-based energy calculations and statistical knowledge derived from the thousands of protein structures that have been determined experimentally over the years, each one teaching the software something about the rules that govern how proteins fold and how they interact with their substrates. In 2008, Baker's laboratory achieved what might be considered a landmark in the field of computational enzyme design, publishing work on what they called Kemp eliminases, artificial enzymes designed entirely on computers and then synthesized and tested in the laboratory. And while these early creations were not particularly efficient by the standards of natural enzymes, they did work, catalyzing a reaction called the Kemp elimination at rates measurably faster than the uncatalyzed process, and this was, in its quiet way, something of a revolution. The Kemp elimination is a relatively simple chemical transformation, involving the removal of a proton from a carbon adjacent to a benzoxazole ring, which then opens to release carbon dioxide and form a product, and it was chosen precisely because of its simplicity, because it provides a clean test of whether designed enzymes can achieve catalysis at all, before one moves on to more complicated and more useful reactions, and the success of these first Kemp eliminases, modest though their rate enhancements were, demonstrated that the fundamental approach was sound. Since then, the field has advanced considerably, with researchers designing enzymes for an ever wider range of reactions, and crucially, beginning to incorporate quantum mechanical considerations into their designs, recognizing that if natural enzymes exploit tunneling and coherence and other quantum effects, then artificial enzymes might be made even more effective by deliberately engineering these phenomena into their active sites. At the University of California, Los Angeles, a theoretical chemist named Anastasia Alexandrova has been pursuing exactly this line of research, developing computational methods to understand how quantum effects operate in metalloenzymes, those enzymes that contain metal ions at their active sites, and using this understanding to design artificial versions that exploit tunneling in controlled and predictable ways, her work representing a kind of quantum engineering at the molecular scale. Metalloenzymes are particularly interesting from a quantum perspective because the metal centers, often containing iron or copper or manganese or other transition metals, have electronic structures that are inherently quantum mechanical, with electrons in d orbitals that can adopt multiple spin states and engage in complex interactions with substrates, and these features make metalloenzymes both powerful catalysts and challenging targets for computational design. Alexandrova's approach involves using advanced quantum chemistry methods to model not just the average behavior of atoms in an enzyme but also the quantum mechanical distributions of their positions and momenta, treating the nuclei themselves as quantum objects that can tunnel through barriers rather than simply climbing over them, and this level of theoretical sophistication, while computationally demanding, is necessary if one wishes to design systems where tunneling plays a significant catalytic role. Her work has suggested that the efficiency of tunneling depends sensitively on the precise geometry of the active site, on distances measured in tenths of angstroms and angles measured in fractions of degrees, and that small changes in these parameters can dramatically enhance or suppress quantum effects, which means that the design of artificial quantum enzymes requires extraordinary precision, a precision that pushes the limits of both our computational methods and our synthetic capabilities. Meanwhile, other researchers have been drawing inspiration from photosynthesis, that most elegant of biological processes, where quantum coherence appears to play a role in the transfer of energy through light harvesting complexes, and in 2019 and the years surrounding it, several groups published efforts to create artificial photosynthetic systems that might harness similar quantum effects, hoping to achieve energy transfer efficiencies that rival or even exceed those of natural systems. These artificial photosynthetic constructs typically involve assemblies of chromophores, molecules that absorb light, arranged in precise spatial configurations on scaffolds made of proteins or synthetic polymers or even DNA, and the goal is to create systems where energy absorbed by one chromophore can be transferred to its neighbors through quantum mechanical pathways, maintaining coherence long enough for the energy to reach a reaction center where it can be converted into chemical form. The challenges here are considerable, for quantum coherence is fragile, easily disrupted by the thermal fluctuations of the environment, and maintaining it for useful periods of time requires careful engineering of the system's vibrational modes and electronic couplings, a balancing act that nature has apparently mastered but that human designers are still learning to perform. One concept that has emerged from this work is what researchers call vibrationally enhanced tunneling, the idea that certain vibrational modes of a molecule can be tuned to promote quantum tunneling, effectively coupling the motion of heavy atoms like carbon and nitrogen and oxygen to the tunneling of lighter particles like protons, and that by engineering these vibrational modes into synthetic catalysts, one might achieve tunneling rates far higher than would otherwise be possible. This approach requires understanding not just the static structure of a catalyst but also its dynamics, the way it vibrates and flexes and breathes at the molecular level, and designing systems where these motions work in concert with tunneling rather than against it, a kind of molecular choreography that represents perhaps the most sophisticated level of catalyst design yet attempted. The potential applications of artificial quantum enzymes span a remarkable range of industries and technologies, from pharmaceutical synthesis, where enzymes could enable the production of complex drug molecules with fewer steps and less waste, to biofuel production, where improved cellulases, the enzymes that break down plant cellulose, could make it economically viable to convert agricultural waste into liquid fuels, reducing our dependence on petroleum. In pharmaceutical applications, enzymes offer the particular advantage of stereoselectivity, the ability to produce molecules with precisely defined three-dimensional configurations, which is crucial because the biological activity of a drug often depends on which of two possible mirror image forms is present, and artificial enzymes designed with quantum considerations in mind might achieve even higher levels of selectivity than their natural counterparts. For biofuel production, the challenge is somewhat different, involving not selectivity but raw catalytic power, the ability to break the stubborn chemical bonds of cellulose quickly enough and efficiently enough to make the economics work. And here too quantum effects like tunneling might offer advantages, potentially allowing enzymes to operate faster at lower temperatures than would otherwise be thermodynamically possible. Recognizing the importance of this emerging field and the need to train a new generation of scientists capable of working at the intersection of quantum physics and biology, the United Kingdom established in 2019 a Quantum Biology Doctoral Training Centre, a collaborative program bringing together universities and research institutions to provide comprehensive training in this inherently interdisciplinary area. The centre, which draws on expertise from across the UK's academic landscape, represents an acknowledgement that the traditional boundaries between physics and chemistry and biology are increasingly artificial when one is working at the quantum scale, and that the scientists of the future will need fluency in all these languages if they are to make progress on problems like artificial quantum enzyme design. Students at the center learn not only quantum mechanics and molecular biology but also the practical skills of computational modeling and experimental biophysics, for progress in this field requires constant dialogue between theory and experiment, with computational predictions guiding experimental work and experimental results refining computational methods in a cycle of mutual improvement. One of the grand ambitions motivating much of this research is the development of room-temperature quantum devices inspired by biology, systems that can exploit quantum effects for technological purposes without requiring the extreme cooling to near absolute zero that conventional quantum technologies typically demand. Natural enzymes, after all, operate at the temperatures of living cells, roughly 37 degrees Celsius in humans, and yet they appear to maintain quantum effects like tunneling and coherence despite the thermal noise of this warm, wet environment, which suggests that there may be principles of molecular design that permit quantum behavior at temperatures far higher than physicists once thought possible. If researchers can understand and replicate these principles, the implications could extend far beyond enzymes, potentially influencing the development of quantum computers and quantum sensors and quantum communication devices, all of which currently face severe challenges related to maintaining quantum coherence in the face of environmental disturbance. The collaboration between quantum physicists and biochemists that is occurring at centers like the one in the UK, and at institutions around the world, represents a kind of intellectual cross-pollination that has historically been associated with some of science's most productive periods, when insights from one field suddenly illuminate problems in another, and there is reason to hope that this particular collaboration will prove similarly fruitful. Of course, we are still in the early stages of this endeavor, and it would be misleading to suggest that artificial quantum enzymes are about to revolutionize industry or that room-temperature quantum computers are just around the corner, for the challenges remain substantial and the path forward is far from clear, but the progress that has been made is encouraging, and the community of researchers working on these problems is growing. What is perhaps most remarkable about this work is the way it requires us to think about molecular design, to consider not just the positions of atoms and the strengths of bonds, but also the quantum mechanical wave functions that describe the probability distributions of particles, and the ways these wave functions evolve and interact with their environments over timescales of femtoseconds and picoseconds. This is design at the deepest level of physical reality, where the classical intuitions that serve us well in everyday life begin to fail, and where we must learn to think in terms of superposition and interference and entanglement, concepts that even physicists find strange and counterintuitive, and yet concepts that nature has apparently been exploiting for billions of years in the quiet interiors of cells. And so the quest continues, in laboratories around the world, where patient scientists run their simulations and synthesize their proteins and measure their kinetics, slowly accumulating the knowledge that will one day allow us to build quantum machines as sophisticated as those that evolution has already produced, and perhaps, in time, even more sophisticated still, though that day lies far in the future, and there is much gentle, methodical work to be done before we reach it. And now we turn our attention to the instruments themselves, to the extraordinarily delicate apparatus that allows scientists to glimpse the quantum world within living systems, and this is a story that unfolds over decades, beginning with the fundamental question of how one might observe events that occur faster than anything the human mind can readily comprehend, events so brief that they exist for mere quadrillionths of a second before vanishing into the thermal noise of molecular life. To understand how biologists came to study quantum effects in enzymes, we must first appreciate the problem of timescales, for the quantum events we have been discussing throughout this narration occur on temporal scales that seem almost absurdly brief when compared to our everyday experience of time, and the challenge facing scientists was not merely theoretical but deeply practical, requiring the development of entirely new ways of making measurements, new sources of light, and new mathematical frameworks for interpreting the signals that emerged from these experiments. The foundation for much of this work was laid by Ahmed Zewail, an Egyptian-American scientist working at the California Institute of Technology, who in the 1980s and 1990s developed what came to be known as femtochemistry, a field dedicated to observing chemical reactions as they actually happen, watching the bonds break and form in what he poetically called the transition state, that fleeting molecular configuration that exists for only femtoseconds before the reaction proceeds to completion. A femtosecond is 10 to the negative 15 seconds, which is to say 1/4illionth of a second, and to put this in some perspective that might help you drift toward sleep, consider that a femtosecond is to 1 second as 1 second is to approximately 32 million years, a comparison that renders the achievement of measuring such intervals all the more remarkable and perhaps also comfortingly abstract. Zewail's approach relied on ultra-fast laser pulses, beams of light so brief that they could capture molecular motion the way a camera flash freezes a hummingbird's wing, and his technique involved sending a first pulse to initiate a reaction and then a second pulse, arriving femtoseconds later, to probe what had changed in that impossibly brief interval. By varying the delay between these two pulses and observing how the absorption of the probe pulse changed, Zewail and his colleagues could construct a kind of molecular movie, frame by femtosecond frame, showing the dance of atoms as chemical bonds rearranged themselves. This work earned him the Nobel Prize in Chemistry in 1999, and while his initial focus was on relatively simple gas-phase reactions, the techniques he pioneered would prove essential for understanding the quantum behavior of biological molecules. Building upon this foundation, Graham Fleming and his research group at the University of California, Berkeley, developed a more sophisticated approach known as two-dimensional electronic spectroscopy, a technique that allows scientists to observe not just when energy moves but also where it moves and, crucially, whether quantum coherence is maintained during that motion. The Berkeley Laboratory, nestled in the hills above San Francisco Bay, became a center for this kind of research, and the specific advantages of two-dimensional spectroscopy lie in its ability to reveal correlations between different energy states, showing how excitations at one frequency might influence or couple to excitations at another frequency, much like a musical chord reveals relationships between its component notes that would be invisible if each note were played alone. The technical implementation of two-dimensional electronic spectroscopy involves a sequence of 3 ultrafast laser pulses, each separated by precisely controlled time delays, followed by the detection of a 4th signal emitted by the sample itself, and the resulting data, when plotted as a function of the various time delays and frequencies involved, produces those two-dimensional spectra that give the technique its name, revealing cross-peaks and diagonal features that encode information about energy transfer, coupling between chromophores, and the persistence of quantum coherence. The timescales accessible to this method span from femtoseconds to picoseconds, where a picosecond is 1/1,000,000th of a second, roughly 1,000 times longer than a femtosecond but still far briefer than any event perceptible to human consciousness, and this range proves ideal for studying the electronic dynamics of biological systems, where electronic coherence might persist for hundreds of femtoseconds while vibrational relaxation typically occurs on picosecond timescales. The distinction between these two processes deserves a moment of gentle elaboration. For electronic coherence refers to the wave-like superposition of electronic states that enables quantum interference effects, while vibrational relaxation describes the dissipation of energy into the thermal motions of the molecular framework, the gradual conversion of organized energy into the random jiggling of atoms that we perceive as heat. Electronic coherence is typically more fragile than vibrational motion because electronic states are more strongly coupled to the fluctuating environment of a biological molecule, surrounded as it is by water molecules and protein side chains all in constant thermal motion. And yet the discovery that electronic coherence can persist for surprisingly long times in photosynthetic systems suggests that biology has found ways to protect these delicate quantum states from the decoherence that would otherwise destroy them almost instantaneously. For studying hydrogen tunneling in enzymes, a somewhat different spectroscopic approach proved necessary, and in 2014, significant advances in two-dimensional infrared spectroscopy, pioneered by researchers including Peter Ham and Martin Zanni, enabled scientists to observe hydrogen tunneling events in real time for the first time. Infrared spectroscopy probes the vibrational motions of molecules rather than their electronic states, and because hydrogen atoms vibrate at frequencies corresponding to infrared light, this technique is ideally suited to following the fate of hydrogen atoms as they tunnel through barriers in enzyme active sites. The two-dimensional variant of infrared spectroscopy reveals how different vibrational modes are coupled to one another, showing for example how the stretching motion of a bond involving a tunneling hydrogen might be correlated with the motions of the surrounding protein framework that gates and promotes the tunneling event. The technical challenges involved in these measurements are formidable and might serve as a gentle reminder of the ingenuity and persistence required to probe nature at its most fundamental levels. Maintaining phase stability between the laser pulses is essential, for any fluctuation in the relative timing or phase of the pulses will blur the signals and destroy the delicate interference patterns that encode quantum information, and achieving this stability requires vibration isolation, temperature control, and optical components manufactured to tolerances measured in fractions of a wavelength of light. The signal-to-noise ratio presents another challenge, for the signals emitted by biological samples are extraordinarily weak, often buried in a background of scattered laser light and thermal emission, requiring sophisticated detection schemes, signal averaging over millions of laser shots, and careful subtraction of artifacts. The samples themselves must be prepared with exquisite care, for biological molecules are sensitive to photobleaching, heating, and chemical degradation, and maintaining them in a functional state while subjecting them to intense laser irradiation requires a delicate balance of exposure times, sample flow, and environmental control. Complementing these optical techniques, neutron scattering experiments have provided unique insights into hydrogen dynamics in biological systems, and facilities such as the ICIS Neutron and Muon source in the United Kingdom and the Institut Laulangevin in France have contributed significantly to our understanding of how hydrogen atoms move within enzymes. Neutrons interact strongly with hydrogen atoms, far more strongly than X-rays do, making neutron scattering an ideal probe of hydrogen positions and motions. And by analyzing how neutrons are scattered by a sample, scientists can determine not only where hydrogen atoms are located but also how they move over various timescales. The ICIS facility, located at the Rutherford Appleton Laboratory in Oxfordshire, uses a spallation source in which protons are accelerated and slammed into a heavy metal target, releasing bursts of neutrons that are then directed toward samples and detected after scattering, while the Institut Laulangevin, situated at the confluence of the Drac and Isere rivers in Grenoble, operates a research reactor that provides a continuous beam of neutrons for experiments requiring high flux and precise energy resolution. Quasi-elastic neutron scattering experiments at these facilities have revealed the characteristic timescales of hydrogen motion in enzyme active sites, distinguishing between the localized vibrations of hydrogen atoms bound within stable structures and the larger amplitude motions associated with hydrogen transfer between donor and acceptor groups. And inelastic neutron scattering has probed the energy landscape experienced by hydrogen atoms, mapping out the potential wells and barriers that govern tunneling probabilities. These experiments complement optical spectroscopy by providing information about hydrogen dynamics on timescales ranging from picoseconds to nanoseconds, filling in the picture of how enzymes manipulate hydrogen motion across the full range of biologically relevant temporal scales. The year 2020 saw significant advances in single-molecule spectroscopy that enabled the observation of quantum effects in individual enzyme molecules, moving beyond the ensemble averaging that characterizes most spectroscopic measurements and revealing the full distribution of behaviors exhibited by a population of enzymes. When millions of enzyme molecules are measured simultaneously, the resulting signal represents an average over all the different conformational states, local environments, and reaction trajectories present in the sample, potentially obscuring the quantum effects that might be most pronounced in particular subpopulations or under particular conditions, and single-molecule techniques avoid this averaging by isolating and observing one molecule at a time, building up statistics by measuring many individual molecules in sequence rather than simultaneously. These single-molecule experiments revealed that quantum effects in enzyme catalysis are not uniform across a population but rather show significant molecule-to-molecule variability, suggesting that the precise arrangement of atoms in each enzyme active site, the specific conformational state, and the local environment all influence the magnitude of quantum contributions to catalysis. Some individual enzymes showed clear signatures of hydrogen tunneling in their reaction kinetics, while others did not, and this heterogeneity provided important clues about the conditions required for quantum effects to manifest, suggesting that enzymes may have evolved to exploit conformational flexibility as a way of accessing quantum-favorable configurations at critical moments in their catalytic cycles. The development of these techniques has transformed quantum biology from a theoretical speculation to an experimental science, providing direct evidence for phenomena that were once considered too fragile to survive in the warm, wet, noisy environment of living cells. And while the interpretation of experimental results remains an active area of research, with debate continuing about the precise mechanisms and magnitudes of quantum effects, the ability to observe these effects in real time has established a foundation for future discoveries. The femtosecond frontier, once the exclusive domain of physicists and physical chemists studying simple systems in controlled conditions, has expanded to encompass the full complexity of biological molecules, revealing quantum effects in the very enzymes that sustain life. As you continue to rest and perhaps to drift toward the edges of sleep, you might find it comforting to consider that the techniques we have described, these elaborate arrangements of lasers and detectors and neutron sources and cryostats, all serve the simple purpose of extending human perception to realms far beyond our natural senses, allowing us to witness events that unfold faster than thought, smaller than imagination, and yet as fundamental to life as any process we might readily observe. And in this extension of perception lies a kind of humility, an acknowledgement that the universe contains layers of activity and meaning that we cannot directly experience but can, with sufficient ingenuity and care, come to understand. And so we come now to a rather interesting chapter in this story, one that concerns not so much the discoveries themselves but rather the careful, patient debates that swirl around them, the sort of scholarly back and forth that characterizes science at its most rigorous, when researchers must weigh extraordinary claims against the demanding standards of evidence that good science requires. For you see, the field of quantum biology, for all its elegant experiments and tantalizing results, remains a territory of genuine scientific controversy, a place where careful skeptics raise important objections and where the community continues to work through the painstaking process of separating what is real from what might be wishful thinking, what is robust from what might be experimental artifact, and what is biologically significant from what might be physically real but functionally irrelevant. The skeptical arguments deserve our attention first, because skepticism in science is not a barrier to discovery but rather its necessary companion, The careful questioning that ensures we do not fool ourselves in our enthusiasm to find something remarkable. Among the most thoughtful skeptics we might consider Paul Broomer at the University of Toronto, a distinguished theoretical chemist who has spent considerable effort examining the claims made about quantum coherence in photosynthesis, asking whether the oscillating signals observed in those famous experiments truly represent what the original researchers claimed they represented. Broomer and his colleagues have raised important technical objections, noting that the interpretation of two-dimensional electronic spectroscopy is considerably more subtle than popular accounts might suggest, and that the oscillating patterns in the data could arise from several different physical sources, not all of which require invoking quantum coherence between different excitonic states as the explanation. The concern, expressed gently but persistently by Broomer and other careful theorists, is that the field may have jumped too quickly from observing certain spectral signatures to concluding that quantum coherent energy transport was occurring and that it mattered for photosynthetic efficiency. They point out that correlation does not establish causation, that observing quantum beating in a spectroscopic experiment does not automatically tell you whether that quantum effect contributes meaningfully to the biological function you are studying, and that considerable theoretical work would be needed to establish such a functional connection. This is not obstruction but rather the ordinary caution of scientists who have seen many exciting claims dissolve upon closer examination, and who understand that extraordinary claims about life-exploiting quantum mechanics require correspondingly careful verification. These debates came to something of a turning point, though perhaps not a resolution, with the publication of a significant review paper in the journal Nature Chemistry in 2018, authored by Jianshu Cao and an international team of colleagues who set out to survey the entire field of quantum effects in photosynthetic energy transfer and to attempt some clarity about what had been established and what remained uncertain. The paper, which ran to considerable length and drew upon contributions from laboratories around the world, represented an effort by the community to take stock after more than a decade of intense activity following the original quantum coherence experiments in photosynthesis. What emerged was a picture more nuanced than either the enthusiasts or the skeptics had painted, an acknowledgement that quantum effects certainly appear in the spectroscopic data, but that their functional significance for energy transfer efficiency remains a question requiring further investigation, that some earlier claims had indeed been too strong, but that quantum mechanics undoubtedly plays some role in understanding how energy moves through these remarkable molecular systems. The Cao paper and others like it brought into focus what has become perhaps the central objection to the entire enterprise of quantum biology, an objection often summarized in a memorable phrase about biological systems being warm and wet and noisy. The argument runs something like this, and it is worth following slowly because it represents serious physical reasoning. Quantum coherence, the delicate phase relationships between quantum states that allow for interference and superposition, is extraordinarily fragile, typically destroyed within femtoseconds or picoseconds by interactions with the surrounding environment, by the thermal jostling of nearby molecules, by the constant random fluctuations that characterize any system at temperatures above absolute zero. The colder and more isolated you can make a quantum system, the longer coherence can survive, which is why quantum computers must be cooled to temperatures colder than outer space and shielded from vibrations and electromagnetic interference. Biological systems, by contrast, operate at body temperature or thereabouts, are saturated with water molecules that constantly bombard proteins and other structures, and are filled with the thermal noise of billions of molecular collisions per second. How could anything quantum survive such an environment? The skeptics ask. And even if it did survive for some brief interval, how could it persist long enough to accomplish anything biologically useful? This is a powerful objection, rooted in decades of experience with the exquisite fragility of quantum systems in laboratory settings, and it cannot be dismissed lightly. And yet, remarkably, theoretical work has begun to suggest that the relationship between noise and quantum effects might be more subtle than this straightforward argument implies, that under certain conditions the environmental noise that one would expect to destroy quantum behavior might instead, counterintuitively, enhance certain quantum processes. This theoretical framework, developed with particular elegance by Martin Plinio and Susanna Huelger at the University of Ulm in Germany, goes by the name of environment-assisted quantum transport, and it suggests that biological systems might have stumbled upon a sweet spot, a Goldilocks zone where the environment is neither so cold and quiet that quantum dynamics proceed without assistance nor so hot and noisy that they are completely destroyed, but rather precisely calibrated to enhance the efficiency of energy or charge transfer through quantum mechanical means. The basic idea of environment-assisted quantum transport requires a bit of careful explanation, though we need not rush through the details. In a perfectly isolated quantum system, an excitation like the energy packet in photosynthesis would spread out in wave-like fashion, exploring many pathways simultaneously, but it might also get trapped in certain states or interfere with itself destructively in ways that slow its progress toward the reaction center. In a completely classical, noise-dominated system, the excitation would simply hop randomly from site to site, losing the advantages of quantum superposition but also avoiding the trapping problem. What Plinio and Huelger showed, through careful mathematical analysis and computer simulation, is that intermediate levels of environmental noise can break the traps and overcome the destructive interference while still preserving enough quantum character to retain some wave-like advantages, creating a regime where energy transfer is actually faster than it would be in either the perfectly quantum or perfectly classical limits. This is a remarkable theoretical insight, suggesting that evolution might have discovered not how to protect quantum effects from environmental noise but rather how to harness that noise constructively, how to work with the warm, wet noisiness of biology rather than against it. These scientific debates, conducted in journal pages and at conferences and in the quiet conversations of researchers puzzling over data, gradually began reaching a broader public through popular accounts of the field. Perhaps the most influential of these was a book published in 2014 by two scientists based in the United Kingdom, John Joe McFadden of the University of Surrey and Jim Al-Khalili of the University of Surrey as well, both of them thoughtful researchers who had become convinced that quantum mechanics played a more significant role in biological systems than had previously been appreciated. Their book, titled Life on the Edge, surveyed the entire emerging field of quantum biology, from enzyme catalysis to photosynthesis to bird navigation to the possible quantum basis of genetic mutations, and presented these ideas to a general audience with clarity and enthusiasm while being reasonably careful about distinguishing established results from more speculative possibilities. The book attracted considerable attention and helped establish quantum biology as a subject worthy of public interest, drawing students and researchers from other fields who might not otherwise have encountered these ideas. As the field matured and the debates continued, the scientific community began establishing more formal structures to support quantum biology research, a sign that the area had achieved a certain legitimacy even if many specific claims remained controversial. In 2020, a new academic journal titled Quantum Biology was established, providing a dedicated venue for publishing research in this interdisciplinary area, bringing together physicists and chemists and biologists who had previously scattered their work across journals in their individual disciplines. The existence of such a journal does not settle any scientific debates, of course, but it signals that the community of researchers working on these questions has grown large enough and coherent enough to sustain its own publication venue, and it provides a forum where the ongoing arguments about evidence and interpretation can be conducted in a focused and sustained manner. Funding agencies, too, have begun directing resources toward quantum biology research, recognizing that even if many questions remain open, the potential significance of understanding quantum effects in living systems justifies investment. The National Institutes of Health in the United States has supported research programs examining quantum effects in biological processes, particularly those related to enzyme function and to the possibility of improving our understanding of drug interactions at the molecular level. The National Science Foundation has similarly funded research in this area, often through programs that bridge the traditional boundaries between physics and biology. These funding increases reflect a judgment by program officers and review panels that quantum biology, despite its controversies, represents a legitimate scientific frontier where careful research might yield important insights, whether those insights confirm the more dramatic claims about biological quantum effects or reveal more modest but still interesting physics. The future applications that researchers imagine for quantum biology, should its more optimistic claims prove correct, span a remarkable range. In drug design, understanding whether and how enzymes exploit quantum tunneling might allow pharmaceutical chemists to design better inhibitors or activators, molecules that interact with enzymes in precisely the ways needed to alter their quantum-assisted function. If proton or hydrogen transfer in enzyme active sites involves significant tunneling, then drugs that modify the shape or electrostatic environment of those active sites might have effects that would be hard to predict from classical reasoning alone, and quantum mechanical modeling might become an essential tool in the drug designer's kit. The enzyme-drug interaction, already complex enough when considered classically, might reveal new dimensions of subtlety when quantum effects are properly accounted for. Artificial photosynthesis represents another area where quantum biology might inform technological development, as researchers seeking to design systems that convert sunlight into chemical fuel with high efficiency naturally look to the photosynthetic systems that evolution has already optimized over billions of years. If those natural systems achieve their remarkable efficiency partly through quantum coherent energy transfer, then artificial systems might benefit from incorporating similar design principles, arranging chromophores in geometries that support quantum superposition and environmental coupling in the ways that biological light harvesting complexes apparently do. The goal of efficient solar fuel production, so important for addressing climate change and energy security, might be advanced by understanding the quantum tricks that plants and bacteria have long employed. Perhaps most intriguingly, some researchers have begun to wonder whether biological quantum systems might inspire new architectures for quantum computing, whether the strategies that evolution has developed for maintaining useful quantum effects in warm, wet, noisy environments might be adapted or mimicked in artificial quantum processors. Current quantum computers require enormous effort to maintain coherence, cooling systems to millikelvin temperatures and isolating them from vibration and electromagnetic noise, And even then, coherence times are often too short for complex calculations. If biology has indeed solved the problem of achieving useful quantum computation at room temperature in aqueous solution, then understanding exactly how it accomplishes this feat might revolutionize our approach to building quantum computers, suggesting new error correction strategies or new physical implementations that embrace rather than fight environmental coupling. And so the debates continue, as scientific debates should continue, with data accumulating and theories being refined and skeptics raising important objections that demand thoughtful responses. The question of whether quantum mechanics plays a functionally important role in biology, beyond the obvious quantum mechanical nature of all chemistry, remains genuinely open, a question that the scientific community is working through with the patience and rigor that difficult questions require. Some researchers remain convinced that quantum effects are central to biological function, that enzymes and photosystems and perhaps even neurons exploit quantum mechanics in ways we are only beginning to appreciate. Others maintain their skepticism, arguing that the evidence remains insufficient or that alternative explanations account for the data without invoking functionally significant quantum coherence. Both positions are held by thoughtful scientists, and the resolution will come not from rhetorical skill but from careful experiment and rigorous theory, from the slow accumulation of evidence that characterizes science at its best, as we drift now toward whatever understanding might emerge from this patient, unhurried, properly cautious inquiry into the quantum nature of life.